It is never a good sign when the schedule gets short. That means the tests are coming. So as you can see, today is our last lecture. In fact, I was looking at the calendar earlier while I was sitting here. We only have, including today, we only have 7 more lectures in this class. Isn't that crazy? We have this lecture, then an exam, then 6 on the nervous system, which again is ridiculous. We could spend a whole semester talking about the nervous system, but you're going to get 6 lectures of it, and then 3 more exams, and then it's done. Then it's Christmas time. Yikes, I know it is scary. But let's talk game plan. For the very last time, our lecture will be muscle physiology. Our labs will be origins, insertions, and actions. We should be able to get through everything that we need to get through. Yeah, well, let me finish this, and then I'll answer your question. I see there's a big question there. And I'm hoping, again, we're still a little behind on the muscles where we wanted to be, so I'm not sure how long it'll take, but if there is time at the end, I will have time. I will, answer any questions you guys have for the exam. And the exam is 1 week from today. We have no lecture on Monday because of the holiday, so I will have my normal office hours except for Wednesday. I won't, it's a holiday, so I will not have my office hours, but I will have office hours the rest of the week. So if you have any questions and things like that, you can email me or come to my office hours. And then we have the exam 1 week from today. All right, so let me see what we wrote here. Okay, great question, Laura. So here is the process. In the breakdown of glucose. So when we have glucose, our job is to break it down to produce energy. And the first step in that process of breaking down glucose is the process of glycolysis. In glycolysis, what ends up happening is that glucose is broken down. Oh, that's a sad, pathetic little arrow. There we go. It is broken down into pyruvic acid or pyruvates. Oops, and more specifically 2. It's broken down into 2 pyruvates. And this process of glycolysis, of breaking the glucose into 2 pyruvates, produces 2 ATP. All right. Now, what happens is, in the presence of oxygen, when there is oxygen readily available, what ends up happening is that those pyruvates enter into the mitochondria. And when they enter into the mitochondria, they're broken down completely. And in that process, we produce lots of ATP, somewhere between 32 and 34. So the total from this process of going into the mitochondria with oxygen and producing ATP is somewhere between one glucose giving us somewhere between 34 and 36 ATP. However, when oxygen isn't available because we've depleted it or things along those lines, without oxygen, what happens is that pyruvate is converted into, that pyruvic acid is converted into lactic acid. And that is where we have that toxin that builds up in that process. So it's not that it is a byproduct of anaerobic respiration. Really, it's more the lactic acid that is the byproduct of it, because without that oxygen, the pyruvates cannot enter the mitochondria. They cannot be broken down completely, and they're converted into lactic acid. That's how, when we were talking about repaying our energy debt, when we repaid our energy debt, one of the things, oops, no, hold on, I want that to be black. When we were talking about repaying our oxygen debt, one of the things that we were talking about is we need oxygen to convert those lactic acids back into pyruvates, and then we need oxygen to take those pyruvates and complete them, break them down into the mitochondria. So part of that oxygen debt, remember, is the breaking down of that lactic acid. Remember, this happens both in the muscle cell, but it also is going to happen in the It's also going to happen in the liver, where we do it that way. All right, so yes, so absolutely, that process of glycolysis is splitting the glucose into pruvates. It's what happens next based on whether oxygen is available or not that determines whether we get a lot of ATP or whether it just gets converted into lactic acid.
Thank you.
In the case, so remember, with oxygen, the pruvates enter the mitochondria. Without oxygen, the pruvates do not enter the mitochondria. And so it stays in the cytoplasm, and in the cytoplasm is where it is converted into lactic acid. If we can get those pruvates into the mitochondria with the oxygen, it'll be broken down completely. But without it, it stays outside of the mitochondria in the cytoplasm, and that's where it becomes the lactic acid. Great questions. Any others? This question is actually a really good lead-in into what we need to talk about next. So any other questions before we move on? All right, well then what we need to talk about next is that everything I've told you up to this point in time is an utter and complete lie. Okay, well not entirely, but one of the things that we have been oversimplifying is our discussion of skeletal muscles. When we talked about skeletal muscle cells or skeletal muscle fibers, we've been talking about skeletal muscle fibers as just one single thing. Although I think we did hint at it a little bit when we were talking about twitches. Remember when we talked about twitches, we saw that twitches in different muscles had different speeds to them. Anyone remember where we had the fastest muscle twitches? Which muscles provided that fastest one? Yeah, the muscles that move the eye. Absolutely, those eye muscles. And then we saw a slower one in the gastrocnemius, an even slower one in the soleus. And the reason for that is there is actually more than one type of skeletal muscle cell. There are actually three main types of skeletal muscle cells. Oops, not calls, cells. Or fibers. Remember, we can use those terms interchangeably. And so when we talk about those, there are several characteristics by which they are different from each other. One of the ways our three main types of skeletal muscle cells are different from each other is in the fiber diameter, the width of the muscle cell. Now notice in parentheses there, I have strength of contraction. Why is that? If I have two muscle cells that are exactly the same length, but one is much thicker than the other, how does that influence and affect the strength of their contractions? Are they both going to have the same strength to
the same time?
I'm sorry?
They contract the same time?
True. Yeah, if they contract at the same time, will they produce the same amount of tension, these two muscle cells that I've drawn?
No.
Why not?
Because one is larger, the second one, because in the larger one, we have a lot of fibers.
Well, be careful. We're not talking, this is a muscle fiber. This is a muscle cell. So you're right. The bigger one is bigger because of what? It has more what in it?
Protein.
Diameter.
I'm sorry, say again?
Diameter.
I thought I heard someone, there you go, absolutely. Myosin, excellent, I like that. Remember, bigger cells have more proteins. Bigger cells are bigger because they have more proteins inside of them, all right? And in particular, the protein we're interested in, as Allison pointed out, was myosin, right? Remember, how much tension a muscle cell produces is determined by how many myosin heads it has grabbing and performing power strokes. And so the more proteins you have, the more myosin you have, the stronger your contraction is going to be. So when we talk about muscle cells that are different in their diameter, That means they have a different amount of proteins, and the more proteins they have, the stronger the contraction. So when we talk about the diameter of a muscle cell, we're also basically talking about the strength of that muscle cell. Excellent. Another way they're going to be different is in the speed of the contractions. Remember, we said myosin heads on average grab and form their power strokes, form those power cycles 5 times a second. All right, they reach, they grab, they pull, they let go. They do that 5 times a second on average, right? That was the key word we used, right? We didn't say all of them do it at that rate. We said on average. And the reason for that is there are some slow myosin heads and some fast myosin heads. And if you have slow myosin heads, what does that do to the speed of your contraction? Slower. There you go. And if you have fast myosin heads, what kind of contraction are you going to have?
Fast contraction.
Fast. See how easy that was? There you go. Exactly. So again, when we talk about the speed of contraction, what we're really talking about is the speed of the myosin heads. Myoglobin. Remind me again what myoglobin is.
Muscles that store oxygen.
Exactly. Remember that special protein inside, the inclusion inside of our muscle cells that allow them to store oxygen. Our 3 main types of skeletal muscle cells vary on how much myoglobin they have inside of them. Some of them have a whole lot of myoglobin. Some of them have very little myoglobin in them. And the same way that hemoglobin binds oxygen by using an iron, and when iron and oxygen come together, it has the red color that we give our red blood cells. The same thing is true for our muscle tissue. Muscle tissue that has more myoglobin is going to have more oxygen bound to that iron on the myoglobin and is going to have more of a reddish color to it. Whereas muscle cells that have very little myoglobin is going to have very little color to it. And so it's more of our white fibers. I know this seems silly to think of muscle with different colors to it, but Thanksgiving is right around the corner. And of course, what is the most important question on Thanksgiving?
Who gets the leg?
True. Okay. Well, there's 2 of those. Absolutely. And I have 2 kids, so it's that way. But yeah, absolutely. More general, right? Light meat or dark meat, right? Do you like the leg? Do you like the breast? Do you like the wing? Do you like the thigh? Right? White meat and dark meat. That white meat and dark meat is white and dark because of the myoglobin content. Right? That's not a debate I want to get into, Haley. People have 2 strong feelings about that. I mean, talking politics is one thing, but I'm not going to talk light meat or dark meat. No way. Way too controversial. All right. Now, another difference in our muscle cell type is their blood flow to them. Obviously, a muscle cell that has a better capillary supply is going to be able to get more oxygen, is going to be able to get rid of its lactic acid if there builds up, right? Going to be able to get more glucose, all those types of things. But really, the main thing about a capillary supply is how quickly can we get oxygen. So capillary supply really is about how much oxygen can we get to that muscle cell and how quickly can we get it there. Well, we'll see. Let's talk about that. Absolutely. We'll see which is which. Absolutely. We will definitely talk about that. Lastly, they're going to differ in the number of mitochondria they have. Some muscle cells are going to have a lot of mitochondria. Some are going to have very few mitochondria. All right. But you got the right idea. If it's all about oxygen, right? Those that are red in color have a lot of myoglobin to store a lot of oxygen, right? Haley, as you pointed out, if you have a large capillary supply, you're going to be able to get a lot of oxygen to the cell. And if you've got a lot of stored oxygen and ability to get a lot of oxygen, you're going to want a lot of mitochondria. Conversely, if you don't have a lot of myoglobin, if you don't have a good capillary supply, you're not getting a lot of oxygen to that cell. And if you're not getting a lot of oxygen to that cell, does that cell need a lot of mitochondria? No, because remember, the mitochondria only work if we have oxygen. if we're not able to get a lot of oxygen to it, then there's really no point in having a lot of mitochondria. So notice what these three factors in particular really deal with is how the muscle cell is going to produce ATP. Those that are getting a lot of oxygen are able to produce ATP aerobically. Remember, these are the ones with oxygen. And those types of muscle cells we call oxidative. Those that don't get a lot of oxygen primarily have to produce their ATP anaerobically, and we call those glycolytic. So in identifying our muscle cells, these factors determine how they produce their ATP or relate to how they produce their ATP. So let's go through our three types and put them together and see what there is to see. So let's erase that. Let's go here. And then actually let's go instead to the whiteboard. So our first type, as we just saw, was fast glycolytic. Fast glycolytic are the most common. The most common type of muscle fibers. Let's make this a little smaller so I have a little bit more room to write. And there's a lot in a name. When we say these muscle cells are fast, what do you think we're referring to? Right, the speed of contraction, absolutely. We were talking about the speed of the contraction. They're going to produce a very fast contractions. And of course, what is going to allow them to produce that fast contraction? Fast myosin heads, exactly. Remember we said there are fast myosin heads and there are slow myosin heads. This one has fast myosin heads. Something else we should know about these fast glycolytic cells are these are the largest in diameter. What does that tell us, that these are the largest in diameter? True, they do take up more space, but what was the other significance to having a large diameter? What gives them that large diameter? What are they filled with?
More
More
protein.
myosin, more protein.
More proteins. And if they have more proteins, what does that tell us about their contraction? More strength, strong contractions. And in fact, being the largest in diameter, they have the strongest contractions. So notice these fast glycolytic muscle cells produce strong, fast contractions. Very powerful, very rapid contractions. But then there's the other half of their name. Glycolytic refers to how they make ATP. And glycolytic means which way do they produce ATP? Aerobically or anaerobically?
Anaerobic without oxygen.
Exactly. They use anaerobic respiration. And as you mentioned, I'll give myself a little bit more room. There we go. That means they primarily make their ATP without oxygen. So if that is the case, do you think they need a lot of myoglobin, or do you think they're going to have very little myoglobin? Yeah, very little myoglobin. Right? In fact, the fact that they have very little myoglobin is why these fast glycolytic cells are often referred to as our white fibers, because they have very little myoglobin to them. All right. These with little myoglobin, do you think they have a large capillary supply or a small capillary supply? Is it as important to get lots of oxygen to it? Yes. So they have a small capillary supply. Small capillary supply. And what about the number of mitochondria? Do you think they have a lot of mitochondria or very little mitochondria? More? Little? Oh, there you go. Excellent. Few mitochondria. Excellent. And of course, as we know, like we talked about, these things that use anaerobic respiration, these things that are not efficient at making their energy, like that Hummer, remember we talked about, needs a very large gas tank. So while it doesn't have a lot of mitochondria, it doesn't have a lot of capillaries, it doesn't have a lot of myoglobin, it is going to need lots of glycogen. Going to need that big, huge gas tank inside of it because it uses that fuel so inefficiently. And while these muscle cells can produce very fast, very powerful contractions, are we going to be able to sustain these contractions for a long period of time? No. So these muscle cells are going to fatigue very rapidly. So these fatigue very rapidly. So what kind of things would these fast glycolytic fibers be useful for?
Short bursts of energy?
Yeah, throwing punches, sprinting, fast bursts of energies, absolutely. Where you get that jumping, right? Something that is very fast, very powerful, but isn't sustained for a long period of time, right? Like we talked about, how long can you sprint for? 10 seconds, 20 seconds, 30 seconds, minute, right? So again, these are going to fatigue very, very rapidly. All right. Questions on that? All right, excellent. Let's talk about our next type. Slow oxidative. Slow oxidative. Again, it's all in a name. It says that these are slow. What do you think that means? What is that referring to? Lipolytic. Yeah, that would be nice. Slow, exactly. Well, so in this case, slow means absolutely the speed of the contraction. And you're right. The reason we have a slow contraction is because it has slow myosin heads. All right. These are actually the smallest in diameter, which means obviously it has the fewer proteins inside of it, fewest. And so, of course, that relates itself to a weaker contraction. So notice these muscle cells produce a much slower, a much weaker contraction than the first type do. However, there's also the second half of their name. So how do these primarily make ATP?
With oxygen.
Exactly, with oxygen. These are going to be the ones that are make it with oxygen. Sorry, let's do it this way. They use anaerobic respiration. Let's be consistent. Pardon me, aerobic respiration, which means that they primarily make their ATP with oxygen. All right. Now, of course, that means that they need to have lots of myoglobin. And in fact, that lots of myoglobin is why these cells are often referred to as our red fibers. Again, you may not have thought of this in these terms, but you're absolutely aware of it. All right, we'll talk about this in a second. Let's just finish the rest of our list. Lots of myoglobin. What about capillary supply? Lots or little? Yeah, lots of capillary or large capillary supply, let's say that. Lots of mitochondria. And so as someone pointed out earlier, while these are producing slower and weaker contractions, we are able to sustain these contractions for a much longer period of time. These muscle cells are fatigue resistant. Again, we were just talking about the turkey. Where is the turkey's white meat? Breasts and wings. Where is its dark meat? Legs and thighs, absolutely. How does a turkey primarily get around? Yes, it waddles, it stands, right? Is a turkey capable of flight? Well, yes, it's not, you're right, Allison, it's not its primary way of getting around, Haley. It's not its primary way of getting around, but absolutely, they can produce flight if they need to in a short burst for a short period of time. How many people here have eaten duck before? Excellent. It's typically when you eat the duck breast is the most common piece. And anybody can describe to me what that duck breast looks like? Yeah, that duck breast is dark meat, because how's that duck's primary way of getting around? Flight, right? It needs to be able to produce those sustained contractions of the wings to produce that flight. It can walk, it can, kick on the water, but it's not its primary way of getting around. It's dark meat, it's in its breast and in its wings. So what are we? Are we turkeys or are we ducks?
Chickens, turkeys.
We're turkeys, absolutely. Chickens are the same way, absolutely, right? We have more white meat in our breasts and in our wings, right? We can produce those powerful contractions, right? As we talked about, we can stand for hours, we can stand for days. How long can you hold yourself in a planking position? Right? Again, when you're planking, you're not even holding up all of your weight. But Haley can only do it for 30 seconds, which is pretty good, right? Better than me, probably. Right? So again, we're not even holding up all of our weight, but these muscles are not set up for that long-lasting, sustained contraction. So we are absolutely turkeys, or chickens, I guess, if that makes you feel better. Either way. And of course, what would we use these slow oxidative fibers for? Yeah, endurance, like we said, standing, right? Walking. Like I keep saying, everything is walking distance if you have time, right? Endurance type slow, sustained activities. Excellent. And that brings us to our last type. The last type of muscle cell are fast oxidative. Excellent. Now, again, it's all in a name. These are fast, and so of course by fast we're talking about the speed of the contraction. And so these being fast, they are going to have fast myosin heads. Now, if fast glycolytic are the largest and slow oxidative are the smallest, what does that make fast oxidative?
The oxygen, the myosin
Well,
head.
okay, yeah, the oxidative part, but I'm just talking size-wise. When we're talking size, if one is already the largest and one is already the smallest, then where must fast oxidative fit? Right,
Medium.
in the middle, absolutely. So medium, or let's use the term intermediate. It is intermediate in diameter. So of course that means it's going to be able to produce more intermediate strength contractions. Excellent. Now, remember the other way we characterize these are how they make their ATP. This is the only one that is a little bit tricky. In this case, while the name is oxidative, what's special about these fast oxidative fibers is that they are equally able to produce ATP, both aerobically and anaerobically. So both with and without oxygen. So they can switch back and forth much more easily than the other muscle cells are. Now, to be able to switch, and again, once again, they're going to be intermediate. They are intermediate in the amount of myoglobin. And if they're intermediate in the amount of myoglobin, how do you think we refer to these muscle cells? They're not red, they're not white, they got to be somewhere in between. Bingo. These are our pink fibers. Absolutely excellent. They are going to be intermediate in their amount of mitochondria. Oh, intermediate in their capillary supply. They are intermediate in their amount of mitochondria. And while they can produce fast and more powerful contractions, the slow oxidative, they're also going to be relatively fatigue resistant. And what type of activities would these be good for?
Middle distance events.
Yeah, absolutely. So again, walking would be slow oxidative, but a jog would be more fast oxidative, right? That would, or a marathon runner would be fast oxidative, or a swim in the pool. And again, by swim, I don't mean like Michael Phelps goes, bursting from one side to another, but a leisurely kind of swim in the pool. Those types of, I like the half endurance. I like that. The half fast stuff. Absolutely. So again, jogging, swimming, things along those lines. All right. So these are our three main types of muscle cells, skeletal muscle cells found in the body. And as we've already kind of hinted at, are these evenly distributed throughout the body? Do we have an even amount of all of these muscle cells all over our body? No, absolutely not. As we just talked about, in our arms, where we can only plank for 30 seconds, you have far more of the fast glycolytic. In your legs, where you can stand for hours, you have more of the slow oxidative. However, almost every muscle in your body is a mix of all three types. Of course, what's the pesky word in that sentence? Almost, absolutely, almost is. You only have one set of muscles in your body that are true muscles. And those are the muscles that move your eye in space. You have 6 muscles that move your eye in space. Thankfully for you, you don't have to learn them for this exam, but I will give you a sneak peek. You are going to learn them for the next exam, for the nervous system. But we have 6 muscles that move our eyeballs through space. Those muscles are 100% fast glycolytic muscle cells. But every other muscle in your body is a mix of all of them. An even mix of all of them? No, like we talked about. In the chest, in the arms, it's going to be more fast glycolytic, right? In our deep muscles of our leg, it's going to be more of the slow oxidative, giving us that posture, allowing us to stand upright. And here's the other thing. Does every single person in this class have the exact same numbers of all of these fast and slow and fast oxidative in our body throughout? No, absolutely not. All right. And again, you might not have thought of it in these terms, but you know this, because if every single one of us worked out every day for the exact same amount of time, doing the exact same amount of reps with the exact same amount of weight, would all of us see the exact same gain in muscle mass and strength as a result of that?
No, genetics.
Yeah, exactly. What primarily determines what number of these you have is genetics. All right, that's why some people who are able to practice, running, get decent at running, where others become world-class sprinters.
I think I'm gonna need you. I'm sorry.
I'm sorry, can you guys give me 1 minute? Minor household emergency. Give me 1 second, please. I'll be right back. So again, I apologize for that. Thank you for your patience on that. So we were talking about how genetics determine how many of these we have in our different things, in our different areas, different regions of our body. And so that obviously some of us are better at faster, more powerful motions like weightlifting or boxing. Others of us are better at slower activities, or jogging or, marathon runners. And again, that's the real key to this as well. You can have 2 world-class athletes like Usain Bolt and Meb, the marathoner, right? And Usain Bolt can't decide tomorrow. Well, he could decide tomorrow that he wants to be a marathon runner. And is he going to be able to instantly become a world-class marathon runner just by deciding he wants to? No, right? He's one of the best runners in the world, best, sprinters ever in the history of sprinting. But he's not going to be able to instantly change, because one of the things that he's been doing is he has been emphasizing with all of his works out and exercises, the fast glycolytic fibers, maximizing the efficiency of those. Genetically, he probably has more of those, which allow him to be more successful in that fashion. Now, does that mean with time he would never be able to become a marathoner? No, you can change these. While this is set genetically, there is some ability to be able to modify this with use, with time. It is possible in particular to convert our fast glycolytic into fast oxidative. So it is possible with a lot of time, with a lot of exercise, endurance type activities, it is possible to convert that. Likely, and again, it also goes by to the side of these. If you, again, both marathon runners and sprinters are world-class athletes, but when you look at their legs look dramatically different, right? The fast glycolytic fibers being the largest in diameter, these sprinters have massive tree trunks for legs, right? Whereas the slow oxidative, the fast oxidative, the endurance muscles are much, much smaller. So marathoners, while they're also world-class athletes, they're not usually the ones that are showing up in the bodybuilder magazines as a result of that. They don't have the same kind of muscle mass as they would as well. I wouldn't say that those are the only ones. It's just the easiest. I mean, obviously, when you're dealing with something that already has slow myosin heads, right, it'd be very hard to convert that entire muscle fiber into suddenly having all fast myosin heads. So changing the speed of the myosin heads is not something that would be easy. Now, again, remember, as we build muscle strength, they get larger, we make more proteins, and it might be possible to add more fast myosin heads to something like that in time. But I think it's easier to get them to at a molecular level, to get them to change how they're producing ATP to be able to build up more endurance that way. All right. Questions on that? All right, excellent. So again, we did this on the whiteboard, but we've got all this stuff here as well. Again, our fast glycolytic are the largest in diameter. There are white cells because of that low amount of myoglobin. Very few mitochondria produce their ATP anaerobically, have that large store of glycogen, produce fast, powerful contractions, but fatigue easily. So again, these are intense movements, weightlifting, throwing a ball, et cetera, et cetera. Slow oxidative are the smallest in diameter. There are red fibers because, again, they have the most myoglobin, produce their ATP aerobically, slow contractions, but fatigue resistant, allows to maintain posture and do other type of endurance activities. And then our fast oxidative, intermediate in color, intermediate in a way that it has the blood supply, things along those lines, because it can equally produce ATP aerobically and anaerobically. Fast fatigue resistance activities like jogging and leisurely swimming. But here's really, oh, not this. Here's the table from your textbook that talks about the three main types. But this is the picture that I really wanted to show you. What we have here is a cross section through a muscle fascicle, a skeletal muscle fascicle. And with this one, what they've done is used a special stain, and this stain stains myoglobin. So notice, as we've talked about, there are some muscle cells that are going to have a massive amount of myoglobin in it. Those are the slow oxidative, the red fibers. Some that will have almost no myoglobin in it, and those are a fast glycolytic. And then here we see lots of muscle cells that have an intermediate amount. Those are our fast oxidative. And notice this is just inside of one fascicle, but if we lower the magnification, and this one, again, using that same stain, we can see that there are three dramatically different types: really dark stained, intermediate stained, and really light stained. So we can see across multiple fascicles, when you take a muscle, as you look in this muscle, and this looks more like a leg muscle because there's a lot of the darker stained ones in here, you can see this one particular one has a lot of the slow oxidative, a lot of the red fibers in it, but they're all intermixed together. All right. So while we've talked about skeletal muscle as being a singular thing, it's really more than that. There are three specific types of skeletal muscle cells. All right, questions on that? All right, with that, then we truly are done with everything we need to know about skeletal muscle cells. We have beaten skeletal muscle cells to death, broken them down to the basic most components, build them back up, and hopefully made some sense of this along the way. What we need to do with the remainder of the class today is to move on, or really move back to our other two muscle tissue types. smooth muscle, and cardiac muscle. Now that we understand what skeletal muscle does, we need to briefly, we're obviously not going to go anywhere near as in depth as we did with skeletal muscle, but we do need to briefly talk about their characteristics, how they're similar, and more importantly, how they're different from skeletal muscle cells. All right, so that is going to be our game plan. It is a little bit early, but this is a good natural stopping point. So let's go ahead and take our first break here. It is 8:48, so let's go ahead and come back at 9:03, and at 9:03 we will restart. I know some of you already snuck out early and got your coffee, but I did not, so I'm going to go sneak and get my coffee, and then we will restart at 9:03, and I will start the recording at that point. And during the break, please feel free to talk more about things that smell good, like Bath and Body Works. All right. Any other questions? All right, see you guys in 15 minutes.
Sir, do we have these topics in the book, like the topics we are talking about?
I don't believe Harvest Moon Pie is in our textbook, no.
No, what I'm saying, not that topic.
Oh, not that topic.
Smooth muscle topic.
Oh, yes, absolutely. Yeah, absolutely, it is in there. All right. Let's go ahead and get started. All right, excellent. So tell me what you already know. Let's talk about this. What do we already know about smooth muscle? Again, way back, did I start it? I think I did. Yeah, I started. Way back at the beginning of this section, which I know feels like 1,000 years ago, especially because it was before the election and so much has happened since then. But tell me what we already know about smooth muscle.
It's uninucleated.
Excellent. Oops. What else do we know?
Is it
Not
peristalsis?
I'm sorry, say again?
I think there's the word peristalsis where they constantly.
Oh, okay, excellent. Yeah, absolutely. So absolutely, we use rhythmic contractions called peristalsis to move substances through our body. Our digestive system, our urinary system, lymphatic system, many of our hollow organs. Oh, I like that. Lines the hollow organs. Excellent. Whoops. What else?
Their shape.
Shape. Okay, excellent. What is their shape? Spindle shape. I also saw that they're involuntary. And so because they're involuntary, how are they controlled?
Nervous system and hormonal system.
Okay, excellent. So definitely hormones. I like that. I'm okay with that. But let's be more specific than nervous system. What branch of the nervous system? Because our skeletal muscle is controlled by our nervous system too. But does what control our skeletal muscle and what controls our smooth muscle the same?
Autonomic.
Autonomic.
There we go. The hormones and by the autonomic nervous system. Excellent. I can think of at least one more thing that we talked about that we knew about them. Let's give myself a little more room.
Regeneration.
Yeah, there you go. Highest rate of regeneration. Excellent. Has that spindle shape. We got that spindle shape. And remember, we talked about, where do we put it? Lines hollow organs. And along with that, it forms layers, right? We see that example here in this nice illustration. Notice that, for instance, in this example, let me switch to my highlighter so I can show you this more clearly. Notice in this illustration of the small intestine, there's not one, but two layers of the smooth muscle. This innermost layer, if you notice, the muscle cells are going around in a circle. They form a circular layer. And as we know, when a circular layer contracts, it changes the diameter. But notice this second layer runs the length of the organ. It is what is called a longitudinal muscle layer. And so when that contracts, the organ gets shorter. So Laura mentioned peristalsis, and that's exactly what happens with these muscle layers. First, the circular contracts, then the longitudinal, then the circular, then the longitudinal. It's the alternating pattern of these firings of these two layers that produce that peristalsis, those rhythmic contractions that move the food through our small intestine. Excellent, excellent. So these are all absolutely things that we have talked about, that we know about our smooth muscle cell. That is spectacular. Excellent. So we have all of that. But here is one of the big keys. It is non-striated. It does not have stripes. And now we understand what those stripes are. We know that the stripes are caused by the precise alignment of proteins into sarcomeres. So if a smooth muscle cell doesn't have stripes, then that means that it does not have sarcomeres. And if it does not have sarcomeres, can it contract the same way that our skeletal muscle cells do?
No.
All right, so exactly. So it must contract using a different mechanism. Now, the good news is I'm not going to make you break it down to all of its tiny bits and pieces and see how it works. But we do need to understand how its proteins are arranged. It's going to use a different mechanism and a different arrangement of proteins to allow it to contract, which is going to produce a very different type of contraction. All right. Now, I'm also not going to save the end. I will spoil it ahead of time. Remember, we had 3 specific types of skeletal muscle. We have 2 specific types of smooth muscle. Those 2 specific types of smooth muscle are what we call single unit, or is also known as visceral. This is the most common type. Most of our smooth muscle is single unit smooth muscle. These are the ones that are arranged in sheets or layers. These are the ones that line our hollow organs. But the reason they're, and which is why they line our hollow organs, which is why they're called visceral, but they're called single unit because these have a large number of gap junctions. So if you think about it, and let's cheat, go back to our whiteboard. And just draw some, now I know these are supposed to be spindle shape, but we will just draw them as rectangles because it takes less time. With these single units, there are those connexons. Remember those special proteins that form the gap junctions connecting the muscle cells together. And as we've talked about, the advantage of gap junctions is that they can work together as a single unit, which is why it gets its name. Because what happens is you have your neuron come in and form a synapse with one of these muscle cells, and it produces an action potential, that big positive signal. And as we know, that action potential is then able to spread to the next cell, depolarizing it, and it forms an action potential. And then that spreads to the next one, and that spreads to the next one. And so what happens is, notice one stimulus to one muscle cell gets all the muscle cells to fire together. And when all the muscle cells fire together, they work together as a single unit. Notice each individual muscle cell doesn't need its own neuromuscular junction, right? Remember, in skeletal muscle, every single muscle cell needed its own neuromuscular junction because each one works independently. And so this is our single unit muscle cells. They align the hollow organs, they form the layers, they work together as one. And in fact, some of these can actually be autorhythmic. Someone remind me again what that means to be autorhythmic?
It does contraction by itself.
Right,
Like
exactly. You've got absolutely the right idea. Autorhythmic means that it is able to produce its own action potential, right? As we talked about, we take that skeletal muscle out and we put it in a petri dish, we give it all the oxygen and nutrients that it needs, and it will happily live there, but it's never going to contract. It can only contract when it is stimulated by a neuron, because that neural action potential produces the muscle action potential. But some of these smooth muscle cells actually can become autorhythmic, where they can produce their own action potential and spread it through. We have a fancy name for this. There's an elaborate network of nerves in our digestive system called the enteric plexus. And this enteric plexus has a more common name. It is often referred to as the gut brain. Oops, so I spelled brain right. What happens is you have that nice big cheeseburger for breakfast, and when you eat that cheeseburger for breakfast, it causes your stomach to stretch. That stretch of the stomach sends a signal to this gut brain, this enteric plexus, and it actually makes its own decisions on how it's going to process this. And then it makes that decision and sends a signal to the stomach, causing the stomach to churn more, causing the stomach to produce and release more hydrochloric acid, and it processes it. And none of that is that's nothing that we have to consciously think of. We certainly don't eat a cheeseburger and go, okay, stomach, now start working on it. But it's not controlled by our autonomic nervous system. Can it be modified by our autonomic nervous system? Absolutely. But it's actually able to make some decisions in some locations by itself. Now, does all single unit smooth muscle do this? No. Right? There's just some special cases where this occurs. But there are some rare instances where that does occur. Now, our second type of smooth muscle is what is called multi-unit. You've actually talked about multi-unit smooth muscle before in this class. We keep talking about how most of our smooth muscle is formed in the walls of our hollow organs. But can anyone give me an exception of that? Where's a location that we have already talked about in this class where we have smooth muscle that is not lining a hollow organ? Where did we find smooth muscle that did not line a hollow organ? There are several examples, but I'm looking particularly for the one that we've already talked about in class. There you go, absolutely. The erector pili muscles. What were those again? In the skin. Excellent. Those are what we call multi-unit. In this case, the muscle cells, and I'll just draw them as lines this time because it's easier. All right. Don't all necessarily have to run parallel to each other. They can just be kind of bundled together. But the real key to this is that for every single one of them gets its own neuromuscular junction. Each one must be controlled independently, more like with a motor unit or those types of things that we see in skeletal muscle, where they're controlled independently, like our erector pili muscles that give us those goosebumps. Absolutely. So those are examples of some multi-unit individual fibers, aren't autorhythmic, don't have gap junctions, and work independently. And again, if you don't like my art, here's the pretty pictures from the textbook that show these two types. All righty. So the good news is, regardless of whether it is single unit or multi-unit smooth muscle, how they contract are going to be the same, and their anatomy is going to be the same. So let's take a look at the anatomy. Here we see a pretty picture of it, but like always, let's go ahead and draw this ourselves. So let's go back to the whiteboard, clear this silly picture, and cheat and use my diamond shape to give us a spindle shape muscle cell. Excellent. Now, as we know, this muscle cell is going to have a large centrally located nucleus. Right, remember our skeletal muscle cell is so chock filled with proteins that the nuclei gets shoved way out to the outside of the cell right underneath the plasma membrane. That's not going to be the case here. We know this is a big centrally located nucleus, but it's going to be in the way of my drawing. So I'm going to go ahead and get rid of it so that we can see some of the other anatomy. Now, as it turns out, our smooth muscle does indeed still have myosin and actin. It has a big, thick myosin filament. But one of the interesting things about this myosin filament is the orientation of the myosin heads. Remember, in a skeletal muscle, all the ones in the right, when they're primed, point to the right. All the ones on the left, when they're primed, point to the left. That is not the case here. On our myosin inside of the smooth muscle, on one side, all of the myosin heads point in one direction. And on the other side, all of the myosin heads point in the opposite direction. Now, we still need this myosin to pull on actin. So there's still going to be actin filaments. For the myosin to pull on. But as we know, it doesn't do any good for the myosin to pull on the actin unless the actin is anchored to something. And in this case, the structure that the actin is bound to is a large protein that is actually connected to the plasma membrane of the cell, known as the dense bodies. So here we have, let's go ahead and do this small, dense bodies. That are going to be the anchor points. Cheat, move this way. Okay, I know this isn't looking perfect, but you get the idea. Cheat and do that a little bit more. Pretend that connects. All right, something like that. Now, we'll finish the rest of the anatomy of this in a second. So we have our myosin, we have our actin. Let's go ahead and put those labels there too. Obviously, we have a sarcolemma. But there are no transverse tubules. And there is very little sarcoplasmic reticulum. Obviously, if that's the case, there are certainly no triads, right? That's obvious in that if there's very little sarcoplasmic reticulum, there's not going to be any terminal cisternae, there's no transverse tubules. But why that is significant is because remember, the sarcoplasmic reticulum is where that skeletal muscle cell stores all of its calcium. We still have regulatory proteins. And we still need calcium to move them. However, our smooth muscle cell relies much more on extracellular calcium. It gets a lot more of its calcium for the movement of the regulatory proteins from outside of the cell. Now, it's still going to need help to be able to do that. And the way it does that is by having these tiny cave-like invaginations on the outer surface of our sarcolemma. And these tiny cave-like invaginations, which is exactly where they get their name, they're referred to as the caveolae. These caveolae are these tiny invaginations that store calcium outside of the cell. So that while it's not being stored inside the cell, there is calcium closely located around the outer surface where it is able to get that calcium that it needs to bring it in. All right. Now, one more anatomical feature. Here we see one example of the myosin and the actin and the dense bodies they are connected to. But obviously, there's not just going to be one dense body inside of this muscle cell. There are going to be many of them. And between these dense bodies, there are going to, of course, be, and my dense bodies are slowly getting larger, but you get the idea. They're all connected to the plasma membrane, but they're all also interconnected to each other as well. Part of the cytoskeletal structure are intermediate filaments interconnect all of these dense bodies together. All right. So this is the basic anatomy of our smooth muscle cell. Now, let's think about what's going to happen here. This muscle cell gets stimulated, it produces an action potential, calcium is going to come inside. We don't have to worry about all of those mechanisms, but I want to see what happens when we move those regulatory proteins. Notice when we move these regulatory proteins, this myosin is going to pull this actin and this, I don't like that, let's make that bigger. This actin and this dense body towards the middle. This one is going to grab this actin connected to this dense body and bring it towards the middle. So notice what's going to happen is our two dense bodies are pulled towards each other. But notice as this dense body is moving in this direction, it's going to be pulling on the dense bodies that are near them. And as this dense body gets moved, it's going to pull on the dense body associated with that. And the same thing's going to be happening on this side. These are going to get pulled this way, and this is going to get pulled that way, and this is going to get pulled that way, and so on and so forth. Notice what's happening here is as this muscle contracts, because of the dense bodies connect to the sarcolemma, and because of these intermediate filaments, what happens is we get this very twisting contraction. Whereas this muscle cell contracts, it does indeed get shorter, but this muscle cell also twists and bulges as it contracts. And again, I know I've done an okay job of showing this here, but let's look at the pretty picture from your textbook. Again, it has the myosin and the actin, but no sarcomeres, no transverse tubules, no sarcoplasmic reticulum. It has the dense bodies. Here we see the dense bodies that connect it to the plasma membrane. And again, are the anchor points for our actin. Intermediate filaments connect the dense bodies together. And even though I don't have it here, again, the caveolae are these invaginations that keep the calcium close by when needed. So when the muscle contracts, the actin is pulled by the myosin. And notice we get this is a great picture. So we have this nice long spindle-shaped cell, but because of this twisting force that is produced by all of these intermediate filaments pulling all of the dense bodies, we get a twisting and a bulging of the muscle cell as it contracts. Now, notice my description of it was very awkward because it is a very awkward activity. Is this going to be fast or powerful with this type of contraction? What do you think? Is this very efficient? Do you think it's going to be super fast? Do you think it's going to be super powerful in what it's able to produce?
No.
No, it is a very slow, very weak contraction. It is in fact about 30 times slower than the contraction of a skeletal muscle cell. But because of the interconnection, only a small amount of energy is needed to sustain this contraction. And so it can maintain this contraction for a much, much longer period of time. Very little amount of ATP is able to sustain this contraction for an incredibly long period of time, which allows us to produce slow but very long-lasting contractions. All right, your alimentary canal, your digestive system, as we've talked about, is a hollow tube from mouth to anus. And in a cadaver, that is about 30 feet long. Why do I have to specify in a cadaver? Don't you have an alimentary canal? Doesn't your digestive system go from mouth to anus? Yeah. But is yours right now inside your body 30 feet long? No. Is it shorter or is it longer? Shorter. Why?
It's contracted.
Yeah. Right? Death is many things, but one of the things that it is very, very relaxing. We have muscle tone in our small intestine. We have muscle tone in our large intestine because this smooth muscle is almost in a constant state of contraction as it's producing those rhythmic contractions, these slow, sustained contractions, this peristalsis and all those things. When we die, it stops contracting and it relaxes. You lose that muscle tone and the digestive system extends as a result of that. So absolutely, while this is a slower and weaker type of contraction, it can be maintained for a much, much longer period of time. All right, and as we talked about, those rhythmic movements we call peristalsis. Nope, and I'm missing a Y in there. Peristalsis. All right. That's what we need to know about smooth muscle. Questions on that? All right, excellent. Then let's talk about cardiac muscle. And again, let us start with what we already know about cardiac muscle. Tell me some of the things you already know about cardiac muscle. Only found in the heart. Excellent. Involuntary. Although we know that that means how it is controlled. So wait, hold on. Before we go any further, let's stick with involuntary for a second. How is this controlled? Is it controlled by the autonomic nervous system? Remember, I take a skeletal muscle out of my cell, I put it in a beaker, provide it with all the oxygen, the nutrients that it needs, and it won't contract. I pull a muscle cell out of my stomach, put it in there and it won't contract. But if I take that heart muscle cell out, it can contract. Ooh, excuse me. You are absolutely correct. Right? Our cardiac muscle is autorhythmic. Right? It produces its own action potential. So you're right. It is modified, not controlled, but modified by hormones and the autonomic nervous system. Excellent. All right. What else do we know? Is it multinucleated or uninucleated? Uninucleated. What else do we know about it? No regeneration.
Semi-striated?
It is striated. And again, this tells us something significant. It is striated, meaning that we know it has sarcomeres. And if it has sarcomeres, do we know how it's going to contract? The mechanism and the process? Yeah, we know how it contracts. It contracts in a very similar fashion to skeletal muscle. Notice very similar. I didn't say identical. Be careful. Is it cylinder shape? No, it has that branching shape. Where a single cell may have multiple branches coming off. We see that very clearly with this one right here. Let's take a look. Notice this cell here. We have a cell that connects to 1 and 2 different other cells in that there. Notice also, and I drew on them right there, there are these squiggly lines where the cells connect. Any idea what the name of that special structure is? Pretty sure we talked about it when I looked at these things histologically. Or you could just cheat and look at the picture on the illustration. Not a Z disc. Intercalated disc. Excellent. Intercalated discs are where the cardiac muscle cells form a junction with each other. And remember, we talked about in that intercalated disc that there were, that's what I wanted to go over. Intercalated discs. I'll write myself a note about that real quick. Have a large number of particular types of cell junctions in them. What are the large types of cell junctions we have in those intercalated discs? Gap junctions, large amount of gap junctions, excellent. And what's the other type? Desmosomes. Excellent. Gap junctions allow these cells to communicate with each other, to work together as one unit. Whereas remember, the desmosomes form those tight anchors that are going to hold the cells together to resist the shearing forces of the major pressure that that heart muscle is producing. Excellent. Did I forget anything? I think that list looks pretty good from what we've talked about so far. I'm trying to think off the top of my hand if there's anything else that we've talked about so far, but I think that is good for what we've done so far. All right, let's take a closer look at it because like I said, because it has the stripes, because it has those striations, it is going to have a similar anatomy to our skeletal muscle. Notice it has myofibrils. Those myofibrils are made up of sarcomeres. Notice we have Z discs, we have M lines, we have actin, we have myosin. Notice we have transverse tubules. right? We have all of those things, but notice when you look at the sarcoplasmic reticulum, notice there are no, I think I say it here, a less extensive sarcoplasmic reticulum, and there is no terminal cisternae. And therefore, no triads. Again, this is an indication that our skeletal muscle, that our cardiac muscle relies on more extracellular calcium to move our regulatory proteins. So its muscle contraction is going to be similar, but it's not going to be identical. All right. So let's talk about this. Here, let's actually do this here. Go back to our whiteboard, clear our whiteboard, and draw a couple lines. All right, let's talk about things we already know. We already know about a skeletal muscle action potential. And if we were to draw that, line, line, we have talked about how this is a very rapid process where we get a rapid depolarization and then a rapid repolarization, and it's gone. And this lasts somewhere around 1 to 3 milliseconds. It is an incredibly fast process. Now, of course, we know the rules. One neural action potential produces one muscle action potential. And what does that one muscle action potential produce?
One twitch.
There you go. So when we have a skeletal muscle twitch, which we know is going to produce the, oops, so I spelled muscle right. The tension, produce the force that is going to allow it to contract. And again, if we were to draw this, oops, wrong button. Black. Oh, and I should, of course, label all of my axes. This, of course, is voltage versus time. This is, of course, time versus tension. And we know that when we produce that muscle action potential, our twitch has how many phases to it?
3.
3. What's the first one?
Latent.
Latent phase. And how does the tension change during the latent phase?
Same. No changes.
No change. Let's pretend that's no change. Excellent. What's the second phase?
The contractile phase, or
And
the
how does tension change there?
increases.
Increases. And then what happens next?
Resting phase.
And how does the tension change? Not resting phase. What happens next?
Decreases.
Well, but it's not called the resting phase. What's the third phase? Relaxation phase.
Relaxation. Sorry. Relaxation.
And how does the relaxation phase change? How does tension change during the relaxation phase?
Decreases.
Decreases. Excellent. All right. So we have this twitch that is produced. And do we remember the timing? How long we said that twitch lasts?
Seconds.
How long?
5 seconds.
Oh, well, not anyway. No, remember we saw like our skeletal muscle of our eye, the fast muscle could be as fast as 10 milliseconds. But remember, like in our soleus, it could go to around 100 milliseconds. All right, so again, it's a relatively fast process, but it can vary in the timing of that. Remember, there was one other key factor in our skeletal muscle. Does a single twitch usually move our body through space?
No.
No, I lift this cup of coffee because I'm able to put multiple twitches together. What did we call that process? Where we put multiple.
Trippy.
Well, no, Treppy wasn't where we put them together. Treppy is where we kept getting stronger ones. We want to get to a fused and unfused tendons, but what do we call that when we're adding the waves of tension together?
Wave summation.
There you go. So it uses wave summation. And remember, it's able to do that because of the short refractory period. What was the refractory period again? How did we define a refractory period?
It can't have another action potential until that period is over.
Excellent. It is the brief period of time where our skeletal muscle cell loses its excitability, loses its irritability. It's that brief period of time where no matter what you do to it, it cannot fire another action potential. And remember, that time was very brief, basically about 1 to 5 milliseconds. And so we're able to use the wave summation to put all these twitches together to get a nice, strong, tetanized, fused, or unfused muscle contraction. All right, no new information there. That is all completely old information that we talked about before, but it's going to help us to relate it to what we need to know about cardiac muscle. So let's talk now instead about cardiac muscle. For our cardiac muscle, it also produces a muscle action potential. But it's a different muscle action potential. So let's draw our graph here again, where again we have voltage. Way too big. Versus time. And what happens is it starts in a similar fashion. We get a rapid depolarization, just like we did in the skeletal muscle cell. However, here, remember, not only do we get a rapid depolarization followed by a rapid repolarization, what ends up happening here in this muscle cell is it has a prolonged depolarization. This prolonged depolarization is what is known as the plateau stage. Where it stays depolarized for a prolonged period of time. And then eventually in time, repolarizes. Now, for this exam, you don't need to know why. We'll get to that when we get to the cardiovascular system in 431. In fact, if you take 431 from me, the cardiovascular system will be the very first organ system we will study. But there is a prolonged depolarization before it depolarizes. This action potential can last over 30 times, almost 30 times longer than our skeletal. So it is a much, much longer process. Now, not surprisingly, if the muscle cell depolarizes for a longer period of time, it is going to contract for a much longer period of time. So again, we have tension over here versus time. And when we look at our cardiac muscle twitch, we see some similarities. There is the muscle action potential is produced. There is going to be a latent period. There is going to be a contractile phase, and there is going to be a relaxation phase. So it has the same 3 phases to it. However, here the timing is around 200 milliseconds. So it stays depolarized for a longer period of time, producing a much longer contraction. Why?
Because of prolonged depolarization.
Well, okay, true. We have a prolonged depolarization gives us a prolonged contraction. But why would we want a prolonged contraction?
Uses less energy.
Sort of, but let's think, what is the function of the heart again?
Pump
Pump the
oxygen
blood.
to the body.
Excellent. By a quick show of hands, how many people here have blown up an air mattress before? All right. And I don't mean the way you do it now, where you plug it into the wall and you push a button. I'm talking about with a hand pump. How many people have used a hand pump to fill up that air mattress? Right? Excellent. Several of you. When you're doing that with that hand pump, do you take a whole bunch of really short, quick pumps of that air mattress, of that pump, to try to get the air into the mattress as efficiently as possible? Is that how you use that air pump? Or do you take the whole stroke of that air pump while you're doing that pumping? Which one do you do? Fast and rapid or big, long pumps? Absolutely, right? Exactly. If you try to do this, the whole thing, it would take forever. It takes forever anyway, but it would really take forever if you did that, because the long sustained contraction of the heart makes the heart a much more efficient pump. So by having a longer contraction, you're getting that more efficient pump of the heart. Now notice one other thing. The muscle contracts, produces tension for about 200 milliseconds, but it also has a refractory period. And the refractory period of our cardiac muscle is somewhere around 250 milliseconds. Now, if it's only producing tension for 200 milliseconds, but its refractory period is 250 milliseconds, are we going to be able to use wave summation here? No wave summation. Is that a good thing or a bad thing? Well, let's think about it. When we use wave summation in our skeletal muscle, our muscle produces this sustained contraction where it tetanizes, locks up in that sustained contraction. Is that what you want your heart muscle doing? Do you want your heart muscle tetanizing? Anybody like to have their heart muscle tetanized? No, probably not. Absolutely not. And so the fact that it has such a long refractory period means it's impossible the heart muscle, and that is absolutely a good thing. We don't want to tetanize. We don't want to lock up the heart in that tetanized contraction. So there's no wave summation. And instead, we just get these slow, sustained pumps of the heart. Now, like I said, when we get to 431, we'll talk about all the mechanisms of these. Also, when we get to 431, we will actually learn that there are actually two specific types of cardiac muscle cells, right? 3 specific, yeah, next semester. Oh, yes, next semester is online again. And my understanding, although I guess it hasn't officially come out yet, but my expectation is that summer will be online as well. They have not officially announced anything. They're working currently on the schedule for summer, and currently they're planning for both. But, and again, this is from no inside information. If I had anything, I would tell you, but I just look at the math of it. If we are 100% online for spring, I don't see any way that they are going to be able to rapidly convert the campus to be on campus for summer. My guess is that they would probably want to use the summer to prepare for fall to get us back on campus again. So that is my hope. But while nothing official has been said yet, my expectation is that likely summer will be online as well. Which again, I know on the one hand is disappointing, but on the bright side, that means that I don't have to wear pants again until August of next year. So that is definitely a good thing. All right. And likely I'll be dead because my wife will have killed me long before then as well. All right. Excellent. So when we get to 431 next semester, we will learn that there are 2 specific types of cardiac muscle cell, contractile cells and our conductive cells. But again, that's more sophisticated than we need to worry about for right now. For right now, I just want you to understand how the contractile cardiac muscle cells work, how they are similar and use a similar mechanism to our skeletal muscle, but have a longer muscle action potential, produce a longer twitch, and have a longer refractory period, giving us a long, sustained, smooth contraction that does not tetanize in our heart, which is a good thing. All right. Questions on that? And I will save this picture and post it onto the board. The Canvas site. All right. Well, with that is everything we need to know about muscle physiology. We are completely done with all of the lecture material we need to cover. So what we need to do now is switch gears now that we're done with the lecture. Yes, and the crowd goes wild, absolutely. But we are not done with the material that we need to cover yet. We still need to get back and finish our origins and our insertions and our actions. So let's go ahead and take another break. It's 9:55, so that means coming back at 10:10. At 10:10, have your red pens, have your blue pens, have your handouts, and we will finish off the muscles of the leg because that is all we have left. And like I said, it shouldn't take us 2.5 hours to do that. So what that means is that once we're finished, I'm happy to take any questions that you have on this material to help you to be successful in the exam. So we should have an opportunity for a review. And then again, throwing a lot of information at you guys, but the good news is that you have a whole week now to study and master this material. And again, that doesn't mean that you get 5 days off and then cram for 2 days. Make sure you're using your time efficiently. All right, let's take a break and come back. And let's actually do this. Let's just switch that, close that. Start here. Perfect. All right, so we'll be back at 10:10, and at 10:10, we will pick up from here. Start the recording at that point. All right, see you guys in 15 minutes. So we left off last time and we were working through our origins and our insertions of the leg. We had just finished talking about the muscle group, the hamstrings, and then we jumped ahead to the gracilis and the sartorius because, and again, this is another great illustration that does a nice job of showing this, how all 4 of these muscles, the semitendinosus, the semimembranosus, the gracilis, and the sartorius all come and insert at that same location that is under the medial condyle of the tibia. But we jumped over our largest muscle group that we are responsible for. Actually, I guess it's equal to the rotator cuff. The rotator cuff is 4 muscles as well, and that is the quadriceps femoris. Quad, of course, meaning 4, so not surprisingly, there are 4 muscles that make this up. The Rectus Femoris and the 3 Vastus brothers, Vastus Lateralis, Vastus Medialis, and Vastus Intermedius. Now, this muscle group is considered an anterior muscle group, and that is primarily because of where it inserts. It inserts on the anterior side of the body, and it has effects like anterior muscles. But as the names tend to indicate, these muscles really wrap around the entire leg on the lateral side, on the medial side, and in between. And where we really see that is when we look at their origins. So let's jump to this and talk about the origins and the insertions of the quadriceps femoris. Now, the quadriceps femoris is a muscle group. And what is the key characteristic of a muscle group?
They all insert the same place?
Well, careful. They all share an attachment point. That attachment point could be the insertion. And in this case, this one definitely is the insertion. But remember, for instance, when we talked about the hamstring, all of those originated from the ischial tuberosity. So they shared an origin. So really the key to our definition is that they share an attachment point. But you are absolutely correct that in this case, that attachment point happens to be the insertion. So let's do that. For all 4 of these muscles, what is the insertion?
The patella to the patellar ligament to the tibial tuberosity.
Well, okay, great. So again, good information there, but let's be precise. What is the bone and bone feature that all 4 of these muscles insert into? What is the actual bone feature that they attach to?
Tibial tuberosity.
Absolutely. The insertion is indeed the tibial tuberosity. All right, perfect. It is indeed the tibial tuberosity. However, because we learned the knee in the previous section, we were talking about the bones, and because it's fairly elaborate in these 4 muscles, notice for these 4 muscles and these 4 muscles only, I may ask you for the insertion pathway. That will be exactly how I asked the question on the exam. And more importantly, being the smart, sophisticated students that you are, if I ask you to identify an insertion pathway, do you really even need to look at the picture to see what it is to know what that insertion pathway is going to be? No, because there's only one insertion pathway I am asking you for. And this is going to be the insertion pathway for all 4 muscles. So it's a little bit more elaborate, but you get to use it 4 times. So now, Laura, can you say it again what the entire insertion pathway is?
They insert into the quadriceps tendon to the patella to the patellar ligament to the tibial tuberosity.
Right, so it's the quadriceps. However, as we also learned, this could also be called the patella tendon. Either of those, or patellar tendon, either of those are acceptable terms for that. Into the patella, and then the patella ligament into the tibial tuberosity. So basically what happens is the insertion of these is just the tibial tuberosity. If I ask for just the insertion, you just have to give me the tibial tuberosity. But we also have this insertion pathway. It starts with all of the tendons. Tendons, of course, go muscle to bone. So all 4 of our muscles, all of their tendons come together and then attach to the patella, which of course is a bone. So the dense regular connective tissue that runs from the patella to the tibia must be a ligament, the patella ligament, to the tibial tuberosity. So here we've both described and also drawn the insertion pathway. And so again, that'll be precisely how I ask it. If I ask for the insertion pathway, I want all 4 pieces. Quadricep tendon or patella tendon, either of those are acceptable, into the patella ligament to the tibial tuberosity. If I just ask for the insertion, you just have to give me tibial tuberosity. Now, if I ask you for the insertion and you give me the entire insertion pathway, I will not mark it wrong, right? Because again, you can give me more information as long as that information is correct, as long as you have the correct information there as well. However, if you say something wrong, if you were to say the bicep tendon into the patella and then the patella ligament into the tibial tuberosity, then I would have to mark that wrong at that point. So again, as I've said many times, and again, it happens on your exam. It happened, the other class just got their exams back recently. As you know, people often lose points. I say this again and again and again. People lose points, not because they don't know the information, but because they don't read the questions carefully. Read the questions carefully so you can answer them correctly. Yes, well, I wouldn't say you guys are smarter, but guys are more sophisticated students. You have proven you can be successful in 430, in the 431 class. They've proven that they can be successful. So I wouldn't say that one class is smarter than the other, but the 431 class is more sophisticated students because they have proven they can be successful in that. They actually, I will say they had, on their digestive system exam, they did really well. Now it was just one organ system. It was a 2-week section, so it was a short section, but I think their average on the lab exam was a 78 and it was a 76 on the lecture. But again, remember, to get into 431, you have to be successful in 430. You have to know how to be successful in 430. So they're starting at a different standard, whereas everybody in here, all you had to do is pass chemistry basically to get in here. And as we know, chemistry is pointless. I wouldn't say that the systems are necessarily easier. I think it really depends on your studying style, right? It doesn't have to be a studying style. It is, if you like memorization, then I think the skeletal system and the muscular system is easier for you that way. If you like more processes, then I think things like the digestive and urinary system are better for that. So I think it's the same class. It's just a continuation. The same way I teach 430 is how I teach 431. I would say the only difference is 431 has a little bit more physiology, whereas this one has a little bit more anatomy. So depending on where people are more comfortable, often depends on which ones they like more. Well, again, I don't think that they're necessarily going any more in depth than we are. It's just there's time for more physiology. Those sections have more physiology. So I think there's more physiology, whereas this has got more anatomy. But hopefully you guys will all be successful in 430, and then you'll get to see that. And again, with the small amount of students that are here, most of the ones that are here, I'm guessing, are going to be successful. The ones that aren't are the ones that are dragging down the average for the rest of you guys and making you depressed. But obviously your individual scores mean something a lot different. All right, excellent. So 4 muscles we have to identify. And the good news is all of these have the same insertion. All of these have the same insertion pathway. Questions on that? All right, then let's talk about each individual muscles, and let's talk about the rectus femoris first. Rectus, remember, is a long straight muscle. For instance, we had the rectus abdominis in the abdominal region. Well, here we have the rectus femoris in the femoral region. We already know its insertion and insertion pathway, so someone give me the origin of the rectus femoris. Again, you guys had the break to get your papers, so hopefully there you go. Anterior inferior iliac spine. So that anterior inferior iliac spine is going to be our origin of the rectus femoris, and I'll just put a big RF there so that we remember that. So we have this muscle, and let's go ahead and with the highlighter, long, straight muscle that comes straight down in here. Remember, we've seen this muscle. One of the things to remember about the rectus femoris, and I will make this smaller now, is a bipennate muscle. So remember what that means. What that means is it has a single tendon down the center, and then it has 2 rows of fascicles that connect to it. This way, we're able to get many, many muscle fascicles onto a small single tendon. And remember, as we talked about, the advantage of this shape of muscle is that it gives us a tremendous amount of power in a very limited space. And that's what we have here with the rectus femoris. Right? After all, the rectus femoris has to move that big, huge piece of meat that is your right leg or your left leg, right? So we were just talking about Thanksgiving and Christmas and all the food and all the pies, right? After the holidays, everybody wants to lose weight, and a quick way to lose 20, 30 pounds is to cut off your leg because it's a big, huge, massive piece of meat. And so it requires a tremendous amount of strength to move it. And so this rectus femoris is one of the primary anterior muscles responsible for moving the leg. Now notice also, as we look at this illustration, how many joints does it cross? 2. So being an anterior muscle, what is the anterior muscle effect on the hip?
Flex.
Flex it. And what's the anterior muscle's effect on the knee?
Extend.
Extends the knee. Excellent. So this flexes the hip and extends the knee. Right? Most of you should have finished your muscle activity handout. What activity did you come up with for the rectus femoris? Or what could you have come up with for the rectus femoris? I don't remember if it was on the list or not, but when would you flex the hip and extend the knee?
Kicking a ball.
Yeah, absolutely, right? Trying to kick a field goal this weekend, right? Yeah, you'd have that big, huge swing of the leg, extending your foot, extending your leg for that big, huge, powerful kick. Right? So this big, huge, one of the strongest muscles of the leg, one of the primary flexors of the leg. We'll see, not the only one. There is going to be one more important one we will talk about, but big, huge, strong muscle with this really important and therefore interesting anatomy to it. All right. The problem with a lunge is you definitely, well, I mean, I guess the very beginning of a lunge, but when you're going forward, you obviously have to bend your knee to be able to step as you're taking that. So you're definitely flexing the hip, but I worry that to complete the lunge, you also have to flex your knee. So that would be the one thing I would be concerned about. Is for
Do
that.
we lose points for these activities? Like if we put wrong activity for the
On the homework assignment?
Yeah.
I did say it was graded for correctness. But obviously, I'll be somewhat lenient with these because, again, the point of it was to be a creative activity. But if I don't make it graded for correctness, then too many people will not put their full effort into it. And so to encourage people to really think about this and put their best effort into it, I made it graded for correctness. But I will provide some leniency. As long as someone gets close to the right idea, then I will probably accept it. All right. All right, because again, remember, I'm not going to ask you what's the muscle that, I'm not going to say on the lab exam, which of these muscles allows you to do a lunge or something like that. However, which arm muscle allows you to do a jumping jack? Could that be a possible multiple choice question? Sure, absolutely. So something like that, but it's not going to be on the lab exam. You need origins, insertions, and actions. I want actions. So don't tell me that it gives a lunge. You got to tell me that it flexes the hip and it extends the knee. All right, now notice the location of the rectus femoris, and we can relate that to the vastus intermedius. Obviously it has the same insertion, so what is the origin of the vastus intermedius?
Anterolateral surface of the femur.
Right, so here on the diaphysis, on the anterior and lateral surface of the diaphysis is our origin for the Vastus Intermedius. Notice if we then draw the line for that one, notice it sits right under the Rectus Femoris. So on our muscle models, on our muscle charts, is there any way to see the Vastus Intermedius if the Rectus Femoris is full and intact? No. So you'll notice on the models, the Rectus Femoris can be removed. You'll notice on many of the illustrations, the Rectus Femoris is cut so that we can see that Vastus Intermedius underneath it. Because no matter how big that Vastus Intermedius gets, it is always going to be under the Rectus Femoris. Now notice one more thing about the Vastus Intermedius. How many joints does it cross? Just one. So it is just an anterior muscle affecting the knee. So what is the one action it has? Extend the knee. There you go. Right? So like when you stomp your foot, right? Where your leg stays straight and your knee is extended. You're extending, obviously this is my knee, my foot, my leg, but you get the idea. This was my knee, that extension of it, that stomping down, right? So if you're a river dancer or you're stopping your feet because you don't get your way, right, you are using that Vastus Intermedius. All right. Now we have two more vastus muscles, the Vastus Medialis. And the Vastus Medialis really is on the medial aspect of the leg. And we see that with its origin. What is the origin of the Vastus Medialis? Excellent, perfect. Those are the two key characteristics. The Intertrochanteric Line. Remember, the Intertrochanteric Line is that raised ridge. Wow, that's a little too big. Is that, oh, here, let's do this one. Raised ridge between the trochanters on the anterior side of the femur. But remember the linea aspera, and we'll draw it as a dotted line, is that posterior bone feature, that raised ridge on the posterior side of the leg. Now, of course, we know the insertion pathway, quadricep tendon into the patella, patella ligament into the tibial tuberosity. So basically what happens here is the fascicles of the Vastus Medialis wrap around the leg and down into that quadricep tendon. So we have these fascicles that are just wrapping around the leg and coming in. And again, I don't want to switch slides because if I leave this picture, we'll lose all these drawings that I'm doing. So we'll do this again and look at all these things. But they all basically wrap around the leg and come in, and it's on the medial aspect of the leg. Still only crosses one joint, and it still comes across the knee in the front along that patella tendon. So it still only has one action. It also extends the knee. Now notice also for the Vastus Lateralis, oh, excellent, perfect. The Vastus Lateralis also originates from the Intertrochanteric Line, also originates from the linea aspera, but the Vastus Lateralis, being a lateral muscle, also originates from the lateral-most bone feature of the hip. And what was that lateral-most bone feature of the hip? Greater trochanter, excellent. So it's also going to be up here as well. Notice it also has, and I'll use brown this time, anterior and posterior origins. So again, it is going to wrap around the leg and down and in on the lateral side. But notice one last thing. Our vastus lateralis, because it's lateral and way up here, is going to be more superior than the vastus medialis. So this one is more superior, whereas the vastus medialis is more inferior. Or really more distal. I'd say just use the right terms. More distal. And this one is more proximal. So the vastus lateralis is more proximal and the vastus medialis is more medial. And I think this is an important distinction to make. There are three things that I want to emphasize again before I leave this picture. Oh, and I messed up, sorry. The first is the shape of the rectus femoris. It has that single tendon with many fascicles that come into it. And that the vastus lateralis is more proximal and the vastus medialis is more medial, more distal. Let's look at the pretty pictures from your textbook and we can see this. Notice here, when we look at the vastus medialis, we can see the fascicles wrapping around the medial side of the leg. And notice it is more distal. Notice the big belly of the vastus medialis is right next to the patella. The vastus lateralis also wraps around the leg from the lateral side, but it starts up here on the greater trochanter, so it's more proximal. And so notice its belly is up higher and not as close to the knee. Here we have that rectus femoris with that bipennate shape. And notice we have to cut a section out to see the vastus intermedius. And make a point of emphasizing these things because, as we know, our huge, massive bodybuilders have the ability to dramatically increase the size of these muscles. And when they do, it can be hard at first to orient yourself when you are looking at this bodybuilder leg. However, there are a couple key things that I want to point out that's going to help you to be able to do it. Right? There's obviously no muscle on the kneecap, so the kneecap is easy to find. And if you can find the kneecap, what muscle has its big belly right next to the kneecap?
Vastus medialis.
Vastus medialis, absolutely. Notice also that makes this muscle over here what?
Lateralis.
Vastus lateralis, absolutely, excellent. And then in between here, this relatively small muscle. Look at how far away it is from the kneecap. This muscle has gotten much, much smaller. This muscle right here looks tiny, but if you look closely, you can actually see the tendon right down the center where you would get that bipennate shape. This muscle here is the rectus femoris. Notice as your leg muscles get massive, like this bodybuilder's, the vastus medialis gets bigger, the vastus lateralis gets bigger, the vastus intermedius underneath gets bigger. But remember, because our rectus femoris has this central tendon, while it gets bigger, does it get longer? No, it's the fascicles that get bigger, and the fascicles go out to the side. So notice the fascicles get thicker, the fascicles get larger, but the muscle itself doesn't get longer. So what happens to the rectus femoris is the rectus femoris is pushed up on the leg. And so notice its distance from the patella gets much, much further away because all the other muscles are pushing it up and out of the way. Of course, no matter how big these muscles get, are we ever going to see the vastus intermedius here? No, we're not going to see the vastus intermedius here. And because we talked about it at the end of last class, let's go ahead and do it again. Notice also, we see this strap-like muscle that is wrapping around the head of the vastus medialis. And what would this strap-like muscle wrapping around that way be?
Gracilis.
Well, notice this one's starting lateral and coming medial. The gracilis, remember, would start medial and stay medial. So the
Sartorius.
gracilis would be there, the sartorius would be there. So we can actually see, I've kind of drawn all over this, but that's okay. Let me erase that one at least. We can actually see that strap-like sartorius coming across the muscle like that. But notice we don't see the gracilis. Notice the inner, even on this bodybuilder, the medial most part of the leg isn't very well defined. Because guess what medial muscles of the leg do? Being a medial muscle on the leg, what's the most obvious action it's going to do?
Adduct.
Adduct, absolutely. Adduct the leg. And do you really need to use any muscle contractions to adduct the leg? If I were to abduct my leg right now, what would I need to do to adduct it?
Nothing.
Nothing. I relax it and gravity is going to pull it down, right? Gravity does most of the work of bringing our legs together. Now, are there machines you can use to tone and work your adductor muscles? Yeah, you go to the gym and they've got these big, huge contraptions that you like, the torture contraptions you get in that you have to use to squeeze your legs together, right? You're not allowed to make eye contact with anybody while you're using those machines, right? Or at least at home, you can get like that Thigh Master from Susan Summers or whatever it was and be able to do things that way. But typically, these are not muscles, even these huge, massive bodybuilders, these are muscles that they don't do a tremendous amount of work to tone. And so they tend to be less defined. So notice we're not going to see that gracilis on the inside. Yeah, you don't make eye contact with anybody and you hope nobody's looking at you while you're on there as well. Absolutely. All right, so while there is a tremendous amount of definition, there's not much on the inside of the thigh. But there we see that long strap like sartorius and those others. So again, if you can find the kneecap, you can pretty much orient yourself with all the rest of these muscles. And that's the nice thing about that vastus medialis. Always be, no matter how big it gets, it's always going to be right next to the kneecap. All right. Excellent. We did the gracilis already. We talked about the fangirl pose. We did the sartorius already, and that leaves us one more muscle of the thigh that we need to talk about, and that is the adductor magnus. The adductor magnus, as you can see, is deep to the gracilis. It is part of a muscle group called the adductor group, which include the adductor longus, the adductor brevis, and the adductor magnus. But I'm not even holding you responsible for the entire muscle group. All you need to know from this group is just the adductor magnus. It is a deep muscle to the gracilis. Let's actually go back a picture. So notice here is the gracilis, and deep and a little bit anterior to the gracilis is where we see the adductor magnus. The adductor magnus, as its name would indicate, is the largest of the three adductor muscles. And how many actions does the adductor magnus have? Three. What are they? Being an adductor, what's the most obvious action of it? Adduct the leg. Excellent. What are its other two actions? Medially rotate the leg and flex the leg. So again, if this is my leg, it would flex it at the hip, right? It would adduct it, bring it in, and it would medially rotate it. Right? I always think of this one as kind of that roundhouse kick. When you're doing that roundhouse kick, you're bringing the leg across. That is what that adductor magnus does. Now remember, as we talked about, like the gracilis being an adducting muscle, it typically doesn't get a lot of tone to it. It doesn't get strengthened the same way that other leg muscles do. The reason I've picked this individual muscle out rather than the whole group is because often in sports, you hear about people getting groin injuries, right? So-and-so's on the ER for a couple of weeks because of a groin injury. Well, I'd say probably about 80% of the time when an athlete has a quote unquote groin injury, it is typically a strain or a tear to the adductor magnus. Because of that lack of tone, when they're running, when they're doing the splits, when they're turning or twisting or doing something that way, this muscle has less tone in it. This muscle tends to get strained. And so again, approximately 80% of all quote unquote groin injuries are injuries to this adductor magnus muscle. And it is deep and slightly anterior to the gracilis. All right. Questions on that? All right, from there, we are done with the thigh muscles, and we can go to the muscles of the lower leg. Now, remember, we have a bicep brachia and we have a bicep femoris. They were similar in name, but were they really similar at all in their orientation or their anatomy? Not really. This muscle, on the other hand, is very, very similar to the bicep femoris. The bicep brachia. Bicep brachia, we have two bellies side by side, a long head and a short head. Here, we have two bellies side by side. And how do we identify the two individual bellies? Be specific. Medial head, excellent. And lateral head. Perfect. We have a medial head and we have a lateral head. Superficial muscle, two bellies side by side, right? Over the top of a bone, the humerus. Here we have two bellies side by side over the top of the bone, the tibia. Superficial muscle. And the same way we do the curls for the girls, do people, boys in particular, care about the size and shape of their gastrocnemiuses? Absolutely, they do. Absolutely, right? You have to have those pretty calf muscles, right? Back in ancient times, and by ancient times, I mean the 90s, for the Cincinnati Bengals, there was a wide receiver by the name of Chad Johnson. Great receiver, huge personality guy. And at one point, he was the one who changed his name. His number was 85, changed his name to Ocho Cinco, which is not how you say 85. But again, that was just him. In Cincinnati, he was, and again, he's one of the best wide receivers in all of football at that time. As a radio stunt, he once raced a horse in a sprint and beat the horse. He was incredibly fast, incredibly powerful. And one of the things he was known for was for having massive calf muscles. In fact, a prosthetic company paid him $500,000 so that they could make a mold of his calves. Because the same way, if you don't like your breast size, you can get artificial breasts. If you don't like your calf size, you can get calf implants. And so they paid him $500,000 to take a mold of his calves so that if you didn't like the shape of your calves, you could get calves that look just like Chad Johnson, Ocho Cinco. Right? So again, it's that nice, big, prominent, superficial muscle that everybody cares about right on top. And just like the bicep brachia, while it sits on top of a bone, it doesn't actually connect to it. Let's actually look. Right? The gastrocnemius sits right on top of the tibia. But what are the origins of the gastrocnemius? What is the origin of the medial head?
Medial condyle of the femur.
Yeah. Now, again, by that, do I mean the big knuckle part? No, the knuckle part, remember, is where it forms the joint. So it is right over the top. In fact, again, if we were actually to hold femurs in our hand, you would actually see the indentation on the bone right above that condyle where the medial head would form its origin. And what's the origin of the lateral head going to be? Yeah, exact same thing. This nice little facet, this fossa, I should say, indentation on the bone right above the condyle is the origin of the lateral head. And what is its insertion?
The calcaneus via the calcaneal tendon.
There you go. Our bone feature that it originates into is the calcaneus, the heel bone, right? So notice, like the bicep femoris that sits on top of the femur but doesn't connect to it, this muscle sits on top of the tibia and fibula for that matter, and does not connect to it. We have these two big bellies, belly 1 and belly 2, that sit on top of the tibia and fibula but don't actually connect to it. They connect from the femur to the calcaneal bone, crossing two joints. Being a posterior muscle, what effect does it have on the knee? Flexes the knee, excellent. Being a posterior muscle, what effect does it have on the ankle? So here's how this works with the foot. Let's set up a good, easy, simple rule. Here are your tibia and fibula. Here is your foot. All right. And as we know, the joint that forms here is that C-clamp formed by the medial and the lateral malleoluses. So that those malleoli, let's change the color of this. Those malleoli are basically the pivot point. If a muscle were to come in behind the malleoli, then when it contracts, it's going to pull the back of the foot up. And what type of action would that be? You would stand on your tippy toes and it would be a plantar flexion, absolutely. If, on the other hand, a muscle came in and attached somewhere in front of the medial and lateral malleoli, then it would bring the front of the foot up. And what type of action would that be?
Dorsiflexion?
Dorsiflexion, absolutely, perfect. Obviously, with this one being a posterior muscle, it is going to bring up the back part of the foot. And so its two actions are that flexion of the knee and plantar flexion of the foot. Now this one, I'm pretty sure you guys had to come up with something for. What did you guys come up with? What's a common activity where you use the gastrocnemius? Now's the perfect time of year for it.
Is it the stand on your tippy toes?
Okay, it would allow you to stand on the tippy toes, but then remember, you also have to flex the knee. So while standing on the tippy toes will let you look over that tall fence, it isn't going to involve the knee flexion. So how can we get that knee flexion involved? I'm not sure I know what MJ means. There you go. Allison's got it, right? Absolutely, right? Again, I have a wife, I have two daughters, I have two female dogs. So of course, this time of year, that means that the only thing that is on TV is the Hallmark Channel, because Hallmark has 485,000 Hallmark Christmas movies that are on, right? And they're basically, as far as I can tell, there are basically three rules in a Hallmark movie. First of all, somebody's dead. There's always a spouse that is dead somewhere along the way. So we've got the tragic story there, right? The second thing is that if they kiss early in the movie, they're going to break up, right? If they have the kiss at the end, then it's happily ever after. And you always know it's happily ever after because of that true love kiss. And what is that true love kiss? That is when they're kissing, where you kick the leg up and you point the toe. That true love kiss is that use of that gastrocnemius for that true love kiss. So that true love kiss at the end of the Hallmark movie, where they kick up their foot and point their toe, You're confused. You haven't seen, you got to watch a Hallmark movie. At the end of the Hallmark movie, what always happens, you have that true love kiss. When you have the true love kiss, the foot comes up, the foot goes out, and then you know it's going to be happily ever after. That is that true love kiss. All right. You're still young, Lena. That's the problem. You don't have to watch the Hallmark Channel. But exactly, every Hallmark movie has the same rules. It's the same things, formula. The only thing they do is they change the name of the town. And but there's always that true love kiss at the end. And that's what your gastrocnemius is good for. All right. Excellent. All right. Now, there is one other thing that the gastrocnemius shares in common with the bicep brachia. And that is that deep to the gastrocnemius, there is another muscle that helps it in its function. Exact same thing we saw with the bicep brachia. Immediately under the bicep brachia is the brachialis that helps in the flexion of the elbow. And it's the same thing here. Notice if we remove the gastrocnemius, then underneath it, and it's been cut here, is this big, broad, flat muscle. We can see a little bit of it peeking out behind the sides. This big, long, flat muscle. But that's where it gets its name. It's a big, long, flat muscle that has to be folded up to put into this small compartment. But it gets its name by the fact that it is a big, flat muscle, like the sole fish. A sole fish is a big, flat muscle that probably a big, flat fish that skims off the bottom of the beach. And the soleus is a big, broad, flat muscle. Now, again, we see it has to be wrapped in here because it's such a small compartment. And we also see that with its origin. Notice the origin of the soleus is what? Is a halibut a flat fish too? I don't know if a halibut is a flat fish, but I know a solefish is a flat fish as well. Cool. Okay, there you go. Those do it as well. Excellent. Well, solephishes are that way as well. So what's the origin of the soleus?
The head and proximal shaft of the fibula and the superior tibia.
All right, so basically it starts over here on the shaft and the head of the fibula, and then basically wraps around onto the proximal diaphysis of the tibia. So it has this kind of U-shaped origin to it. And what is its insertion? There you go, into the calcaneus via that same calcaneal tendon. I know I just drew this. Take a quick look at this, but I want to go back to the picture. First, real quickly, how many joints does it cross? One, just like the brachialis. It just crosses one, and that is, of course, the foot. But notice when we look at this, we see that both the soleus and the gastrocnemius use this tendon, this calcaneal tendon, to attach to the calcaneus bone. Now, our soleus, because it only crosses one joint, the ankle, can only do one thing, and that is, of course, the plantar flex of the foot. So you guys were talking about standing on your tippy toes or looking over the fence. That is what our soleus does. Notice these muscles, the gastrocnemius and the soleus, right, are what allow us to push off with our toe as we walk, as we run, as we jump. And this calcaneal tendon is the tendon that has the most tension in it of any tendon in our body. Has anybody here had the unfortunate opportunity to tear their Achilles tendon? Been around someone who has? Have you ever been around in close proximity to someone when they've torn their Achilles tendon? If you in the ER, yeah. So if this happens, were you there, Katie, when he did it? Yeah, part of it does take forever to heal. Absolutely. No. If you're in close proximity, and again, hopefully this doesn't happen to you, but if you're in close proximity, you can actually hear it. You can hear it from like 100 yards away. It is super loud. There's so much tension. It's this popping. And in fact, when you talk to many people who have done a complete tear, they believe they got shot because there's a loud popping noise and then they collapse to the ground because there's so much tension in this. When it tears, that releases that tension, the muscle bunches up into the back of the leg and has to be pulled out. And it can be incredibly hard to heal from. Again, it's a tendon. So it's a dense, regular connective tissue, which we know is poorly vascularized. When it is damaged, it is typically damaged by a cut or a tear that goes perpendicular to it. So again, it's very hard to get all those collagen fibers to reform with any kind of strength or support to them with a complete tear. And often it takes years, 1 to 2 years, to heal as a result of that. And in many cases, the person is never back to the strength and mobility that they had before. Right. So again, it can be a very devastating injury when it occurs. Now, we keep using the term calcaneal tendon, but notice also it is also referred to as the Achilles tendon from Greek mythology, where Achilles was held by his foot into the river Styx to become immortal, except on the one place where his mom held him. And Achilles tendon is such a ubiquitous term. I will allow you to use the term Achilles tendon for identifying the calcaneal tendon. You may use either of those two terms to identify this structure. Again, it is a big white thing. So on the test, if I point an arrow towards there, I would ask you to identify this structure. All right. Questions on those? All right, excellent. One more muscle we need to know the origin and the insertion of, and that is the tibialis anterior. As the name would indicate, the tibialis anterior is an anterior muscle of the lower leg. But reach down and feel the anterior part of your lower leg. Do you feel a lot of muscles in that area? Oh, I don't want to know what that is, Christopher. What did you just show us? Is that somebody popping their Achilles tendon?
Yes, I was curious and had to pull it up. So
yeah, I'm
in case you're curious, in case anyone's curious, there you go.
I've had 3 knee surgeries, so I'm a little bit of a wuss when it comes to the leg stuff. So I'll pass on watching that. But yeah, anybody else who wants to, you're welcome to do that.
It's not gross, but you definitely hear it.
Oh, it's gross for me. I feel it. I feel those kind of things when I see it. All right, but like I said, I'm a knee wuss now, so leg wuss when it comes to that now. All right, excellent. So reach down and feel the anterior compartment of your leg. And one of the things that most sticks out most prominently is that you feel bone. You feel that diaphysis of the tibia. So there are not a lot of muscles in the anterior part of the compartment of the leg. And so the primary one is this tibialis anterior. Now, let's take a look at its origin and its insertion. So I want to actually see if I can increase the magnification of this a little bit. This out of the way, this out of the way. All right, I may have gone a little too big, but that's all right. We'll start here. Oh, I want to get it all on field of view. There we go. Okay, that's a little better. All right. Again, I know I'm not on the right one, but it'll serve our purpose. All right, here, I'll cheat and put it here. We'll do the right one. Okay, so if you think about it, this needs to be on the anterior part of the tibia. However, as we know, is there going to be room on the tibial tuberosity, which is the most anterior and obvious feature, for yet another muscle to attach? How many muscles are already attaching to the tibial tuberosity? 4.
All the quadriceps.
Yeah, all the quadriceps femoris muscles, right? How many muscles are inserting into the medial condyle over here on the tibia?
So medial head of the gastrocnemius.
Now, remember that's up on the medial condyle of the
Femur.
femur. Yeah. How many muscles insert into the medial condyle of the tibia?
3.
Close. Nope, 2's the wrong direction. 4. What are the 4 muscles that insert into the medial condyle of the tibia?
Semimembranosus.
What else? Well, if the semimembranosus does, then of course, what else does?
Sartorius.
Sartorius. What else?
Semitendinosus.
Semitendinosus.
And
Gracilis, absolutely. All right, those 4 all remember insert onto the medial condyle. So notice there's not a lot of space here on the anterior side for our tibialis anterior. So the obvious location for where that origin of the tibialis anterior would be over here, lateral to the tibial tuberosity on the or under the lateral condyle of the tibia. So because it's really the only place for it up there. There's so much other stuff connecting up here. This is really the only place for it. So notice it originates on the lateral side of the tibia. But where does it insert? Where does the tibialis anterior insert?
Medial cuneiform and first metatarsal bone.
There you go. On the medial cuneiform, which remembers this, and the first metatarsal. But it actually does it on the inferior part. It's actually underneath. If this was my foot, this basically, the tendon wraps around and attaches to the underside of the foot. So onto the medial cuneiform, onto the first metatarsal of the foot. It comes up underneath like that. So it's on the underside of it. So notice this muscle is indeed an anterior muscle that basically snakes across the front part of the leg. Now, obviously, cross is just one joint, the ankle joint. Being an anterior muscle coming in front of the medial and lateral condyles, what is its obvious action going to be? Is it going to bring the toes up or is it going to bring the toes down?
dorsiflexion.
All right, bring the toes up, which is a dorsiflexion. But notice, because it wraps around to the front of the foot, it's able to pull the foot so that the foot turns inward. So as it twists the foot, it can pull the foot so that the sole of the foot points inward. And what type of action would that be? There you go, inversion. So the 2 actions of this muscle is to invert the foot and to dorsiflex the foot. All right. Questions on that? Now, I know you may not have thought about this muscle before, but many of you may have without actually thinking about it. Because this muscle is really the only muscle on the anterior part of the leg, as we have talked about, there are fascias that wrap around and stabilize muscles that do the same job. Well, this is the only muscle that does this job. So there is a fascia that wraps around this muscle, holding it and anchoring it in place. And fascias are made up of dense, irregular connective tissues. And are dense, irregular connective tissues super elastic and springy and stretchy? No. So if you were to, for instance, get off the couch after sitting there for the 6 months that we've been here so far and decide to go run 10 miles, one of the things as you use your muscles is those muscles increase with blood, right? They swell from irritation and from the damage. They become enlarged. They become inflamed. And as they swell, if they are held in place by that fascia and can't stretch and can't expand, then the muscle becomes congested with blood. And now as every step you take, it feels like that muscle just wants to rip off your body. What do we call that condition? There you go. Allison's got it. Shin splints. Shin splints often occur because of the constriction of the tibialis anterior by its fascia. One of the easiest ways, the best ways to try to get rid of shin splint issues is to stretch significantly before running. When you stretch, you help to loosen that fascia, and it gives more room for the expansion and the swelling of the tibialis anterior. Now, in some individuals, that shin splints become so bad that no amount of stretching can resolve it. And in some cases, again, it's not a common procedure, but in some instances, if a person is dealing with chronic shin splints, one of the things they will actually do is go in and cut that fascia. If they go in and cut that fascia, that can loosen it up, and that can actually provide some relief for that issue, for that problem. Like I said, it's not the common way that they try to resolve that issue, but it can sometimes be used. All right. Questions on that? All right, that is the last muscle we need to know the origin and insertion of, but you'll notice there are two more muscles here on the leg we need to be able to identify. The first one is this muscle right next to, oh, that's way too big. The first one is this muscle right next to the tibialis anterior. Again, just looking at the belly doesn't tell us a whole lot about it, right? But if we look closely and follow its tendon, we see that its tendon branches to go to the digits of the foot. Didn't we have a muscle in the hand that did something like that? What did we call the muscle with the tendon that split to go to all the fingers?
Extensor digitorum.
Extensor digitorum. Well, this one also extends the digits, but it has a much longer tendon. And because it has a much longer tendon, while in the arm we have the extensor digitorum, what do we have in the leg?
Extensor digitorum longus.
The extensor digitorum longus. Excellent. Now, with a name like extensor digitorum longus, what is its obvious action?
Extension.
Extends the toes, absolutely, right? And because its tendon comes in front of the medial and lateral condyle, is it going to bring the front of the foot up or bring the back of the foot up?
Front of the foot.
So what is its other action?
Dorsiflexion.
Dorsiflexion. So notice this one brings your foot up and brings your toes out. Right? This is the one when you step on something really gross, right? You lift your foot up, move away from it, you extend your toes, right? Because you're all grossed out by that slimy thing that you stepped on or whatever it was. So that's that extensor digitorum. And notice right next to that, here's the extensor digitorum again. And again, its tendon tells you what it does. Right next to that, on the lateral aspect of the leg, is a small bellied muscle with a very long tendon. What do you think we call this small bellied muscle on the lateral aspect of the leg? Small belly, long tendon, lateral part of the leg.
Volaris.
Fibularis longus. All right. or peroneus longus. It's also called that because remember the lateral aspect of the lower leg is also known as the peroneal region or the fibular region. Both are acceptable terms. So fibularis longus, peroneus longus, both are acceptable terms for it. Now, it's a lateral muscle on the lateral side. So again, we don't need to know its origin and insertion, but it's lateral. So when it pulls on the foot, it's going to rotate the foot so that the sole of the foot points outward. What kind of action would that be?
Eversion.
Eversion, absolutely. That is going to be an eversion. And notice for its second action, we need to know how it relates to the hinge, the lateral and medial malleoli. Notice in this case, this one comes, its tendon goes behind the ankle joint, behind the lateral malleolus. Pardon me, the lateral malleolus. So since it goes behind it, it is going to bring the heel up. And when it brings the heel up, right, that is the one that points the toes out, right? And that is that plantar flexion. So the two actions of this muscle is to plantar flex the foot and evert the foot. All right. Questions on that? All right, excellent. And just that simply, we are done with all the material you are responsible for this next exam. All right. Questions on that?
I have a question.
Yes.
So I know on the lab portion you're going to be showing us pictures and all that.
Hmm?
Is it going to be half and half based upon like actual bodybuilders and then pictures as though like the muscle shown, or how is it? Because I just want to know, because it's kind of difficult for me to locate the bodybuilder ones, but not like the.
No, great. I totally great question. That's an excellent question. So as I mentioned, and again, so in the classroom, this is a 30-station exam, and only one of the stations has bodybuilder pictures. So typically on this exam, there is usually about 3 to 4 bodybuilder questions. So that's what I would expect. This test usually has somewhere between 80 and 85 questions. Okay. So of those 80 to 85 questions, only 3 or 4 of them are going to be bodybuilder pictures. So the bodybuilder pictures are definitely not a huge part of this, right? Again, it is a useful activity, but it's not going to be the emphasis. Remember also, as I said, there are bones on this. And in the classroom, usually there's somewhere between 6 and 9 stations that have bones on them. So that's somewhere around 20, 25 questions. So my guess is somewhere around 20 questions. And again, these numbers I'm just throwing out, but they give you an idea of the sense. Less than a quarter of the exam should be bone pictures, right? So somewhere between 15 and 20 pictures, probably bone pictures. The vast majority are indeed going to be pictures of models, pictures of charts of the actual muscles. Right. And on those actual muscles, like the pictures from your textbook we've been looking at, again, you're going to need to be able to answer those 4 questions: origin, insertion, action, and of course, the name of the muscles. So okay. And then also remember, there's also going to be microscopy as well. We've got those great models of the skeletal muscle cells. There's the microscopic slides that we need to be able to identify. In fact, when we come back for the break, there's a couple histology things I want to quickly look at. I think we did it, but I want to make sure that we look at and we're comfortable with that. So I want to make sure we do that. Things that way as well. But so there would be the models and histology slides that you're responsible for, that you have that list of as well. But easily 50% of the test, if not more, is going to be pictures of models or pictures of charts of the individual muscles. Oh, and we missed a muscle. How do we miss a muscle? Here's where we missed it. So when we transitioned from the hamstring to the gracilis, we missed a muscle. There's one more muscle group we forgot to talk about. That muscle group is I thought I had a picture for this. Oh, I know where I have the picture for this. Hold on a second. Now I remember. It's all coming back to me. do this together. All right, let's do the last muscle first, and then we'll look at the pictures as part of the kind of the start of the review, although we will still take a break. The last muscle you are responsible for is a muscle group. That muscle group is known as the iliopsoas. The iliopsoas is a muscle group comprised of 3 muscles. Well, 2.5 muscles. Every single person has an iliacus. Notice the iliacus is a muscle where this muscle is found inside the fossa of the ilium, your pubic bone. Everybody has a psoas major. Psoas major is a big, large muscle where this muscle attaches to the spinous processes of the lumbar vertebrae. And many people have a psoas minor, a small additional muscle that is sitting on top of the major. Although for some people, the major and the minor fuse together. For our purposes, we don't really care because you don't need to know any of these individual muscles. You just need to know what they form collectively. And collectively, they form one of the strongest muscles in our body, the iliopsoas. Notice the iliopsoas comes underneath the inguinal ligament, and then the fascicles actually fuse together, where they all come together to become one indistinguishable muscle as they pull through there. And that iliacus crosses the hip. As we talked about, the hip is this big, huge, massive chunk of meat. It's big and it's heavy. So we have this big, huge bipennate muscle that is the rectus femoris that helps us to move it through space, a big, strong muscle. But here we have a muscle that's actually anchored inside the abdominal pelvic cavity, anchored to your pelvis, anchored to your lumbar vertebrae, big, solid, weight-bearing structures that come and attach to the front of the leg to flex the leg and bring it through space. The model shows this really nicely, but we also see, the illustration shows this nicely, but we also see it really nicely on the models as well. In your modules on Canvas, I have these leg pictures because I wasn't satisfied with the leg pictures that were shown on our that Summits Riveter site. So we talked about some of the things here. Again, notice here we see the short belly of the bicep femoris and the long belly of the bicep femoris. Here we have another muscle I forgot too. Yikes. We have that nice long iliotibial band that we see along the right side here. And we have one of the smallest muscles with the largest names, this tiny muscle right here. Anyone know what this tiny muscle right here that I also forgot to identify and talk about might be? Well, look at your list. What haven't we talked about yet?
The tensor fasciolati.
The tensor fasciolati. Remember, the iliotibial band is a thickened portion of a big, huge fascia that wraps around the entire leg, keeping the entire leg in place. It basically forms that pressure cuff to help to keep blood flowing back to the heart, stabilizing this in place. So there's this fascia that wraps around the whole thing called the fascia lata that the iliotibial band is a part of. And guess what the tensor fascia lata does? Well, keeps tension on the fascia lata. Its job is to help to keep tension on this, to help to provide that pressure cuff, to help to keep the blood going back towards the heart. Now, it is also, as you notice, an anterior and lateral muscle of the leg. So being an anterior muscle of the leg, what is it going to do to the hip?
Flex.
Flex. And being a lateral muscle, what's it going to do to the leg?
Abduct.
Abduct. So now again, is this tiny little muscle going to be the primary abductor or the primary flexor of the leg? No. but it is a prominent muscle that is there. All right, excellent. Have I missed anything else on our list? I got those two. I don't think I missed anything else, but I didn't think I missed these two either. Is there any other muscles, leg muscles on the leg? This is the way we get for splitting it weirdly the way we did before. Any other muscles here we didn't talk about? All right, so we see these nicely on a model like that. Obviously, the large, large prominent gluteus maximus, we can see that there. We can see the gluteus medius sticking up here laterally from this lateral view. So a lot of nice things from this lateral view that we can see. Here's a posterior view. And remember, here in this posterior view, we can see some of the muscles have been removed. So the primary nice muscle that we can see here is the short head of the bicep, the bicep femoris. The short head of the bicep femoris is right there. Notice here we can see how the two bellies of the gastrocnemius are connecting up to the top part of the femur really nicely from there as well. So we can see that really, really nicely. We can also see the gracilis and the sartorius coming in from the side, although not the best view of that, so not likely I would use for that on the exam. Here we see the medial view. So notice with the medial view, we have our sartorius, we have our gracilis. Whoops, hold on, let me be more careful with that. Gracilis, remember the gracilis is superficial, and here, deep to it, deep to the gracilis is that adductor magnus. This one here is the adductor longus, but remember, that's not one of the ones you're responsible for. So deep to, right, posterior and anterior, deep to the gracilis is that adductor magnus. So we see that really nicely here. Again, that big belly of the vastus medialis right next to the kneecap. We can see that. And remember also, we could see the deeper semimembranosus and the superficial semitendinosus here. So again, we can see that these four muscles, 1, 2, 3, and 4, all are coming together. All their tendons are coming together into that medial condyle of the tibia. But I believe this is, no, I lied. Yeah, well, this one works. Notice here from an anterior view, we can see a couple things. Our sartorius from that superior anterior iliac spine. And notice, even though we don't need to know the origins and insertions of it, if we can find the sartorius, we can find the tensor fasciae lati. And in between them, here's our rectus femoris. Notice we can clearly see that tendon down the center and its bipennate shape. No, I will not be asking left and right on this exam. If you haven't figured left and right out by this point, then I've given up hope. No, it's not as important on these because, again, most of the models that are made are only left-sided or right-sided, so it's not as, so left and right's not going to be important on this one. Notice here we see that structure that is the inguinal ligament. But notice also here we can clearly see that muscle group we just finished talking about. Here is the iliacus on the inside of the ilium. Here is the psoas major and minor coming from the lumbar, and they come together and come under the inguinal ligament. That is that psoas, iliopsoas. And notice the other way we can see the iliopsoas really nicely is actually on the torso model. Notice on the torso model, when we look on the inside of the torso, again, we can see the iliacus that is on the inside of the pelvis. We can see the psoas major and minor, and we can see how they come out underneath the inguinal ligament. So this is a muscle that anchors to the core inside the body for that big weight-bearing flex of the leg. Big lift that leg up. So we have this big, huge anchor point on the pelvis, on the lumbar vertebrae to flex the leg. All right. So yes, I would say easily half the test, if not more, will be pictures like this or pictures from your textbook where I will point at a muscle and identify the muscle, iliopsoas, identify the one action of the muscle, flex the hip. And again, being the sophisticated students that you are, it's going to help you on this exam Because if a muscle has 4 actions, how many actions am I going to ask for? Not a trick question. 4 actions. So if I point at a muscle and say, identify the 4 actions of this muscle, is there any chance this muscle is the iliopsoas or the rectus femoris or the vastus medialis or the vastus lateralis or the vastus intermedius? No. I can probably count on one hand how many muscles have 4 actions. And if I'm asking for 4 actions, it has to be one of those 5 or 6 muscles. I can think of 4 off the top of my head, 5. I can think of 5 off the top of my head. There might be others, right? External oblique, internal oblique, sartorius, gracilis, trapezius. Am I forgetting one? Is there another muscle that has 4 actions? I'm trying to think off the top of my head.
Semimembranosus and semitendinosus?
No, they just have 3, don't they? Flex the hip, flex the knee, and laterally rotate. Right, bicep femoris only has 3, pectoralis only has 3, latissimus dorsi only has 3. I think there's only 5. I may be wrong. There may be 1 or 2 others. But the point is, if I'm asking for 4 actions, that tells you something. And remember also when you're answering these questions, don't just say that it flexes or extends or abducts or whatever it is. You have to either tell me the part of the body that moves or the joint. So if on the exam, I point here and ask you to give me the 1 action of this muscle, you can't say to bend over at the waist. What you need to say is that it flexes the hip or it flexes the leg. All right? Give me the action, give me the part of the body that moves or the joint that moves. And read the questions carefully. On the exam, you're going to see this picture with this arrow and you're going to get all excited and you're going to go, I know that's the iliopsoas. And you're going to write the iliopsoas. And the problem is, the question is, what is its 1 action? All right, and so make sure you read the question carefully so you can answer it correctly. And what's the other big 2-word phrase you should watch out for on this exam?
Be specific.
If you see the words be specific, then that tells you one thing right away. This is a multi-headed muscle. Basically, the only time I'm going to use that term be specific. And so I'm asking for about a multi-headed muscle, and you are either going to have to give me the specific origin or the specific name, right? Because they have the same insertion, they have the same actions. So when you see those 2 words, be specific, you know you're either doing an origin or a name, and you know you need to be precise. Yeah, Haley, it really is that. Read the questions carefully. So many people lose points, not because they don't know the information, but because they don't read the questions carefully. You have to read the questions carefully so you can answer them correctly.
Will there be anything like asking about the hamstring or quadricep altogether, or will it always be specified on each individual muscle?
Great question. Absolutely. So on the exam, could I point and have a circle around or even better, doesn't even have to be a circle. Could I, for instance, here's a good picture. This is the perfect example for this, because as I talked about on the exam, there are 4 main questions I will ask. Identify the muscle, be specific. What would your answer to that be? Anyone? Identify the muscle, be specific.
Long head of the bicep femoris.
Close, short head. Remember, this is the short head because identify the origin, be specific.
Linea aspera.
Of the femur. Excellent. Identify the insertion.
Head of the fibia and lateral condyle of tibia.
Okay. Identify the 3 actions.
Extend and laterally rotate the thigh and flex the knee.
Identify the muscle group this muscle is a part of.
Hamstring.
There you go. So that's likely how I would ask the question. I guess I could have a circle around multiple or arrows on multiple muscles and ask you to identify the muscle group, or I could ask you identify the muscle group that this muscle is a part of. So yes, knowing your muscle groups are one of the things that you need to know, yes. Now remember, if it's something like the zygomaticus, I'm just gonna ask for the muscle, right? Because we're treating the muscle group like a muscle, so all you'll have to say is zygomaticus. It's only when we have to know both the muscle group and the individual muscles that it becomes an issue. All right. Where are we? Thought I had some histology pictures. So I will tell you what, let's go ahead and take one last quick break, and then we'll come back and do our official review. And during that review, that'll give me a chance to get some 2 histology pictures in particular that I want to look at, or at least I'll see if they're on the CRC site that we use. All right. Any questions? So again, that's all. Now we are done with all the material you're responsible for the exam. So let's just take a quick 10-minute break, come back at 11:45, and at 11:45, I will answer any review questions you guys have. So again, we will return at 11:45, and I will start the recording for the review at that point. All right. See you guys in about 10 minutes. All righty. So before we get into your questions, as I mentioned, there are a couple quick things that I wanted to show you. I wanted to remind you again, if you're not taking advantage of it, that there are these study tool websites available for you in the modules. And in particular, there are 2 that I use the most when I'm going through these materials, and I think that are really useful. The first is, of course, that virtual anatomy lab that Cosumnes River College has. And what's nice about this is they have many of the similar models and things along those lines that you're responsible for. If we look at histology, again here, what tissue would this be? Anyone know what histology, if this was question 1 on the lab exam, which it easily could be? Skeletal muscle, excellent. In a longitudinal plane of section, right? We can clearly see the striations from that, the nuclei pushed to the side, everything that goes along with that. And remember, with this one as well, not only could I ask you to identify this tissue, but I could ask you for characteristics. So tell me some of the characteristics you know about this tissue.
Multinucleated, striated.
Excellent. What else?
Cylindric.
So excellent. What else?
Voluntary.
Voluntary, excellent. Controlled by the somatic motor nervous system, excellent. Give me at least one more.
Generation is
yeah, it has sarcomeres and limited regeneration, excellent. All right, so we could do that. Here's a closer look at that. Identify the tissue here.
Smooth muscle.
Smooth muscle. Give me some of its characteristics.
uninucleated,
Uninucleated.
voluntary.
There you go.
Highly regenerative.
Highly regenerative.
Shape is sphere.
Spindle, right? That's a
Spindle
spindle shape.
shape, yeah.
Perfect, excellent. And then here, right, we have cardiac muscle. We can see the branching shapes, but I think I actually like this one a little bit better. This one you can see a little bit better, a couple things. You can see the striations of them a little bit better. But the other thing that you can see really nicely on this one, as we were talking about today, is those intercalated discs. Those intercalated discs are the locations where these come together. And as we said, those are where the junctions form between the two cardiac muscle cells. And there's a massive number of desmosomes and a massive number of gap junctions between them that allow these to work together as one. The other thing that we've talked about and we saw in the models, but I don't remember if we did it as a, I thought this had this. Now I'm confused. Oh, no, I guess it was the other one. Okay, excellent. So that's good for that. The other site that I think is excellent for histology is this Yale histology site. This Yale histology site is really, really amazing. So notice if we go to the muscular system, all these keywords where you can get the vocabulary of a lot of great information and material. You can actually quiz yourself with a pre-quiz. There's actually a quiz mode where you can take this, but you also have a study mode where you can look at these things. So notice again, if we look at our cardiac muscle cells, this one, I think even better than the other two, does a nice job of showing the striations and showing the intercalated discs that we can see in there. Right, so again, it talks about the tissue and things this way. The other thing, if you've looked at your histology list that we are responsible for, is the neuromuscular junction. We have talked about it, but you can actually see it under a light microscope. Notice here, we have our axon terminals that are coming down and branching. And then they branch to form our synaptic end bulbs. Now notice they have this labeled as the motor end plate, but really what you're seeing here is the synaptic end bulb that is sitting over the top. Now we know that synaptic end bulb is the neural specialization, and we know it's sitting right on top of the motor end plate. So we know that those two things come together. And do they touch? No. What do we call the space in between?
Synaptic cleft.
Excellent. And what is the name of the chemical that is released for communication here, the specific name?
Choline. Acetylcholine.
Exactly. And what's the generic term we would use for that?
Transmitter.
Neurotransmitter. Excellent, right? So again, on this histology, we can really just see the synaptic terminals going and the axon terminals into the synaptic end bulbs, but we know all the components of that that are there. And what's really cool, I wouldn't use this on the exam, but I like it. But I won't be on the exam. But it's really, really cool when you're looking at this. Notice they actually have a neuromuscular junction in electron microscopy. So notice in the electron microscopy, we can actually see that synaptic end bulb with the vesicles. We can see the invaginations of the synaptic cleft. We can see the muscle cell with its invaginations. I mean, we can even see the little bit of a sarcomere in here. It's super cool. So again, it's just that all those things we've been talking about, it really actually in there. But so this is an excellent site if you haven't started using it already for the histology. I know this one doesn't have a ton of histology, but when we get to the nervous system next, there is going to be a ton of histology there. So if you're not using these resources already, please start. So that is something that can help you with that. All right. So again, those are the things that I wanted to show you because I think that was the last little bit we hadn't really fully finished talking about yet was the histology. We did the muscle tissue at the very, very beginning, but like I said, that feels like a long time ago. And I don't think we actually did the neuromuscular junction. So I wanted you to remember that you are responsible for that histologically as well as on the models. All right. All right. So that is everything I wanted to cover. So what can I answer for you guys for a review? Remember, if you don't ask questions, I assume you've all mastered the material and I make the tests harder. Everybody's already in vacation mode. You got a day off, so you're already all checked out already. I talked about pie earlier and Thanksgiving to make you guys giving you summeritis. All right. Well, if you guys know this stuff, then excellent. I look forward to grading the material.
I do have a question.
Yes.
In terms of aerobic and anaerobic respiration, can you give an example of a question on what that would be?
Well, so obviously those are two methods that we use to make ATP, right? Those are the two main methods we use to make ATP. So when we're talking about the resources that are necessary for our muscle cell to be able to produce a contraction, we know those two resources are calcium and ATP. So do we need to talk about how we regulate how much calcium is released into the muscle cell? Absolutely. We can talk about depolarizing it, open those voltage gated calcium channels, letting it out of the sarcoplasmic reticulum. And the more that comes out of the sarcoplasmic reticulum, the more calcium we have, the more regulatory proteins we can move, right? From an ATP standpoint, how long or how powerful a contraction we produce is determined also by how much ATP is available. And ATP is used up. It is a resource that is used up. The calcium isn't used up by that process, but ATP is. So remember, we talked about basically 4 ways that we get the energy necessary to contract a muscle. 2 of them involve starting with stored energy already in the muscle, stored in the form of just ATP and stored in the way of creatine phosphate. But stored energy is only enough inside a muscle cell to sustain a contraction for 20 to 30 seconds. If we want to contract a muscle for longer than that, then we have to make new energy. And there are 2 ways to make new energy. Aerobically, which is very efficient, produces a lot of ATP, but is relatively slow. And anaerobically, which is quick and easy, but is very inefficient, only gives us a little bit of ATP. So could I ask you to compare the 2 different ways that you would make ATP? Sure. Could we talk about all the ways you get the ATP necessary for a muscle contraction? Sure. All of those are things that could possibly be a potential essay question. All right. Haley, I saw your question as well, but we'll get to it in a second. Anything else on aerobic versus anaerobic? Well, and now also now after today too, remember we know that different types of skeletal muscle cells use different types of energy making. So needing to know which type of energy or how, fast glycolytic cells primarily make ATP versus how slow oxidative primarily make ATP is also going to be important. All right, so any other questions on ATP production?
I have one question on the neuromuscular junction, not on ATP.
Okay, excellent. Well, tell you what, Haley asked her question first in the chat. So let me answer her question about the contractile cycle, and then we can talk about the neuromuscular junction. So what was it that you wanted to know about the contractile cycle, Haley? Oh, and Max, remember, this is college. You're adults. You're not required to be here. It helps if you want to do well on the exam, but the nice thing about being a college teacher is that I don't have to babysit. You guys are adults. Leave whenever you want. All right. So, okay. So let's go through it together. When, like all of our parts, when we talk about the 3 stages of our muscle contraction, it's always good to review what the goal is. So what is the goal of the contractile cycle?
Convert.
Say again?
Remove the regularity protein and we're using calcium.
Is that what the contractile cycle needs to do? All right, contractile cycles, remember, where we're generating the tension, where we're producing the
Oh, sorry, yeah.
Absolutely, excellent. All right, so where does that begin? I think that's where you were getting confused, Joed, because where does contractile cycle begin? Yeah, contractile cycle. Now remember, careful, we want to be careful. Contractile cycle begins not with the decision, that happened way before. Contractile cycle, when we're just focused on the contractile cycle, it begins when the calcium moves those regulatory proteins out of the way. So troponin rotates the tropomyosin out of the way, and now our actin is exposed. All right, at which point it's a cycle, so there isn't really a stopping point. It's a continuous process, but there are basically 4 processes that have to occur in the contractile cycle, right? The reach, the grab, the pull, the let go. So those are the 4 things that have to happen. So we could really do them in any order that we want, but let's do reach first. What is the reach? What do I mean when I say reach? What is happening in my sarcomere when I say reach? What's occurring in that event?
The myosin head splits ATP.
Yeah, exactly. Before you can grab and pull, you have to be able to get into that primed position. So when I say reach, we're talking about getting into that prime position. And absolutely, Allison is correct. What our myosin head does is it takes an ATP and it rips off the phosphate. So it's holding the ADP and the phosphate, and it is storing that energy. And when it has ripped the phosphate off of the ATP and it has stored the energy, that puts it in that primed position. And how do we know a myosin head is in a primed position?
It's like cocked.
Right. And what direction?
Away from the
See
end
line?
line.
Away from the M line. Exactly. It is in that cocked position where it is pointed away from the M line. Excellent. So that is the reach. Now, once it's primed, it is able to grab. And of course, we're not going to use the term grab. Grabbing has a fancy name. And what does it mean when the myosin grabs onto the actin? What do we call that? Forming a cross bridge, absolutely. So it binds to that actin, forms that cross bridge. As soon as it binds, what happens next? The pull. Of course, again, we need to use fancy words. It's not a pull. It's going to pivot. That power stroke, exactly. That myosin head performs the power stroke. That power stroke is indeed a pivot. We want to describe the power stroke. It's a pivot towards the M line. And what happens when it pulls, when it pivots towards the M line while it's holding on to the actin? Baby step it for me. When it pulls on the actin, what way does it pull the actin? It pulls the actin towards the M line. And what's the actin connected to?
The Z disc.
Z disc. So when you pull the actin towards the M line, you pull the Z disc towards the M line. And when the Z discs get pulled towards the M line, what happens to my sarcomere?
Shortens.
And when all of the sarcomeres in a myofibril all get short at the same time, my myofibril gets shorter. And that myofibril is connected by proteins to the sarcolemma. So when my myofibril gets shorter, what happens to the muscle cell? It gets shorter. When it gets shorter, it's pulling on its connective tissues inside the fascicle. So when my muscle cell gets shorter, what happens to the fascicle? Fascicle gets shorter. And since that fascicle is connected to the muscle cell by the epimysium, when the fascicle gets shorter, what happens to the muscle?
The size of the muscle gets
shorter. It shortens. So notice that contraction, that power stroke of the myosin head ultimately leads to the change in shape of the muscle, the production of the tension that moves our body through space. All right. But that was just one power stroke. One power stroke's not going to help me to lift my cup of coffee to get my caffeine. So I got to keep the cycle going. So the next step is that release, right? We've reached, we've grabbed, we've pulled, but now we need to release. How do we release?
Break the cross bridge by releasing the ADP.
Close. You've got the right idea. Remember what happened, and I know I'm going to end up marking myself when I do this. I'm going to regret this the second I do it. But I split that ATP and phosphate, right? I mean the ATP into ADP and phosphate, and I've stored the energy. That's what the myosin head has done. It stored those and then it primed. Then it grabbed, and then it performed the power stroke. Remember, during the power stroke, not only is it using the energy, but it also releases the ADP and phosphate. So it has an open binding site. And now that it has an open binding site, a new ATP can come and bind to it. And when that new ATP comes and binds to it, it lets go of the actin, right? Just the binding of a new ATP allows it to let go. Now, to keep the cycle going, I have to prime this myosin head again. And luckily, I can rip the phosphate off this new ATP, and that will prime it. And then the process continues, right? And I did get highlighter on the floor, but I don't care. It's all for science. So there you go. Basically 4 continuous steps: reach, grab, pull, let go. Reach, grab, pull, let go. But if I don't tell my wife, who's going to clean it? Me? Nah. My belly's too big. I can't bend over. All right. Exactly. I will, because I don't want to get in trouble. I've already cleaned it up. I rubbed my foot into it till it went away. So now it's on my sock. So it'll be all right. All right. Excellent. So there you go. We have those 4 steps, right? Prime the myosin head, form the cross bridge, perform the power stroke, break the cross bridge, and then prime again. And that process will continue until one of those regulatory proteins moves back in the way. Once the regulatory protein is back in the way, then when the myosin head, that prime myosin head, goes to grab again, it can't, because the regulatory proteins are in the way, and the process stops. So like I said, for me, I remember it by the reach, grab, right, pull, release, but don't use those terms. Use the correct terms. We prime the myosin head, we form the cross bridge, we perform the power stroke, we break the cross bridge, and then we prime the myosin head again. All right? But it really is just like pulling on a rope or climbing a ladder. You reach, you grab, you pull, you let go. You reach, you grab, you pull, and let go. And just like climbing a ladder, you do with different hands at different times. And so it's a continuous process. All right. Someone had a question about the neuromuscular junction.
Yeah, this was me, sir.
So what's your question about the neuromuscular junction?
So
Wait, hold on.
actually,
Before we switch, any other question? I think we covered it, but I just want to clarify. Any other questions on the contractile cycle? Any other confusion on that before we move on? Okay. Now your question about the neuromuscular junction.
so according to the explanation, like the note we took from the lecture and explanation, like just as I've seen the video for the neuromuscular junction and explaining 7 sample steps, that these are the steps happening in the neuromuscular junction. So like in step 1, it explains an action potential travels to the length of the axon and a motor neuron to axon terminal. So as you explained, like it's step 1 in the video. It doesn't say like it makes the axon terminal more positive or polarized. It doesn't use these terms.
Okay. Well, I didn't write the video. I didn't make the video. So while it shows the overview of the processes that what happened, what I am looking for in the description is what I want, the details that are necessary. So that video that you're watching is a great illustration of it. It's a great 3D model that shows that. It implies some information though. It implies that when the synaptic end bulb depolarizes, it does say the next thing that happens is that the voltage gated calcium channels open and the calcium enters into the synaptic end bulb. Yeah. So it does mention that. What I want from you though is the why. Why does that voltage gated calcium channel open? Exactly. And as you pointed out, the reason for that is it depolarizes the threshold. It has a switch that causes it to open. And when that action potential comes down, it depolarizes it to threshold, and that's what opens it. So the one thing that is lacking from that video is some of the whys. But those whys, those details are how you maximize your points on the exam.
Sure.
Yeah.
Thank you so much. Yeah. So this was my goal.
But yeah, I mean, ultimately, again, my goal isn't just to parrot the information from your textbook. So while obviously the videos, the textbook, the other resources do a nice job of explaining these things, I try to express things to you the way that I want them expressed back. So the descriptions and the details that I'm looking for. So again, these other resources are useful, but if you base your answer solely on that video or some other resource, then you may be missing some of the specificity of detail that I'm looking for. And so that's going to be the key to that.
Thank you, sir.
Yep. Great question. Any others? Well, I was about to say that I think Maxim gets his wish by leaving early, but I think he left early. Took that whole being in college and being able to make your own decisions to heart, apparently. All right. Any other questions before we finish up? All right. Well, you have a whole week to study this material. I have all my normal office hours, except for Wednesday. Wednesday, I will not have my office hours because it's a holiday. But I'm also great at responding to emails. I have some plans this weekend, so I may have some availability if the normal office hours to be able to meet outside of that. So, but if nothing else, I can still communicate by email, and then we can always make arrangements to meet if it's easier to discuss things that way. Yeah, I'll try to get it uploaded today as I usually do. I know, shocking it is. I know, and like anybody gets a life while we're in quarantine, but yes. So between birthdays and holidays and everything else that's coming up, it's a busy weekend. But next weekend is more busy. So and I'll be grading your tests as well. So, but the point is, reach me by email if you have any questions, or come to my office hours, and I will help you with that. Guys, be safe, be careful. As long as you spell acetylcholine esterase correctly, if you want to describe it as the Pac-Man that breaks apart the acetylcholine or eats up the acetylcholine, I would have no problem with that. All right, but you just got to spell acetylcholine esterase correctly for that. All right, guys, have a safe weekend. Take care, and good luck on the exams.